have finally reached the sessions that we have planned, the penultimate session and the last session, and this is the last day of this 5-day faculty development program. A very good morning to all my dear participants and my guest today, Dr. Kutubuddin Ali Mullah. So on behalf of our university, Maulana Abul Kalam Azad University of Technology, West Bengal, I extend my thanks and gratitude to our honorable speaker, and as well as this 5-day faculty development program, as you all know, is an initiative by AICTE, Training and Learning Academy. So in brief, we call it ATAL. So ATAL Academy has sent out, has invited many such programs across the country. And we have been fortunate to hold this particular FDP on the applications of CRISPR-Cas gene editing tool, which is, of course, you all have seen how important it is in genome editing and how it can be translated for the societal benefit. So, of course, it has been a very, very fruitful. If I consider this faculty development program, for the last 4 days, we had 12 very excellent sessions, and we have 2 more to go. So I hope all the participants have been able to gather as much as knowledge and the know-how of applying this not only for their own research, but also to teach their students this new technology. And this is the idea, and this is the objective of such programs, because finally you have to take the message from these sessions to your for improving your career and your professional development. So I think we have been able to at least make some contribution towards that end. So, and of course, I will thank all the persons who have been helping me out on this, holding this program. Especially, I will thank my Honorable Vice Chancellor, Professor Dr. Shoykot Moitro, who has encouraged me from the very beginning to hold this program amidst his busy schedule. I hope he will be there in our valedictory session. And of course, all my friends, my colleagues of my department, from my department, my staff, the admin, and everybody. And more importantly, all the speakers who have gladly accepted my invitation. So I am very, very thankful to all of you. And of course, the entire proceedings have been carried out by Prem Kumar. So thank you very much, Prem. You have been doing an excellent job, untiring effort by you. And of course, Shohini backing up in this effort. And we have actually been able to get many intermittent feedbacks also. And I'm happy that it has been a fruitful program at the end. What makes me happy is that whether the purpose of this faculty development program has achieved or not. There can be shortcomings, there can be some demerits in my holding this. I don't think it is very important at the moment that we bring about all the negative, but I will definitely want all of you to be present at the valedictory session so that you all can share your candid views on this program, which will help us to improve in the near future. So I welcome again all of you to this penultimate session, and I take this pleasure and honor to introduce today to the speaker, Dr. Kutubuddin Ali Mullah. And it is going to be another very good session because Dr. Kutubuddin is from the plant background. So he's a scientist in the crop improvement division of ICAR Institute, National Rice Research Institute, Cuttack. So I find that Dr. Kutubuddin is connected to Kolkata. I'm very glad that he obtained his PhD from Kolkata, University of Calcutta. And then he moved on to US at Pennsylvania State University for his postdoctoral research. And of course, he obtained the Fulbright Scholarship also for that. The most important aspect of his research is that he is very much interested in genome editing using this CRISPR-Cas technology, especially for the crop improvement. But that is very, 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 very important. And he has been awarded several awards, like the Young Scientist Award by the Indian Science Congress Association, the Jawaharlal Nehru Outstanding PhD Thesis Award by ICR. Congratulations also on that note. And the INSA Medal for Young Scientist 2020. So from the Indian National Science Academy. That's really wonderful. So your achievements are really great, and I congratulate on you, Dr. Kutubuddin. And also he has received this American Association for Advancement of Science, the Program for Excellence in Science, that award even. So that's quite a few which is worth mentioning, and I'm very proud and very happy that we have you today to deliberate on your topic of research and to the audience. Of course, Dr. Molla has also many publications. He has co-edited books from Springer, and it is very great to see, very encouraging to see that he has been selected as the assistant features editor for the Plant Cell Journal. So whoever is interested and works in plant will know how important it is. And of course, as this faculty development program is focused on CRISPR-Cas gene editing tool. Today, Dr. Kutubuddin is going to talk on CRISPR-Cas and overflowing toolbox for plant biotech innovation. So I think all of you must be very excited and curious, because yesterday's plant session was very encouraging, very interactive, and I want all of you to be actively participating in today's session. So once again, thank you, Dr. Kutubuddin, for accepting our invitation, and welcome to this FDP. The stage is yours now. Thank you.
Thank you very much for your wonderful introduction of the program and of me. But I was a little bit, it was too much large introduction of me. It was not needed, not needed.
No, we need to, because audience have to know that what are the achievements and all. Though they have been sent out, but it is our great honor and pleasure.
Yeah, thank you. So let me just. Okay, so is it visible, my screen?
Yes, it's visible. Absolutely fine. Yes.
Okay, great.
Perfect.
So as you mentioned, the objective of this program, a faculty development program. So am I audible clearly?
Absolutely, very clear.
Okay, great. Because I normally used to use headphone while delivering a lecture, but this morning I forgot to take my headphone in my office. So I'm directly talking, that's why. No. Okay, so the main objective of this program, as mentioned by Dr. Jaya Bandhopadhyay, that to outreach about this emerging and this wonderful technology to as much as possible people or stakeholders who are interested in this. So yeah, it's my great pleasure to be here and to talk about a little bit of my work and what is going on in the plant biology field, harnessing the power of these wonderful tools. And as a member of Indian National Young Academy of Science, that is shortly called as INIAS, so we have another added responsibility other than doing research is to do a lot of outreach. And this is a program, that's why it gives me immense pleasure that this is also a great extent of outreach, because there are a lot of teachers who would take these messages to their students, and it will help them to propagate and help us to propagate the power of this technology. So this is wonderful, and thank you, organizer, for organizing this program. I thank Dr. Jaya Bandyopadhyay for inviting me and for interacting with me throughout this invitation and all this abstract and title. And as she mentioned, Prem Kumar Shoini, I also thank, and obviously Dr. Soikat Mitro and Dr. P.P. Lahiri, all of you. Thank you for inviting me. So let me just show you a figure here. This is in front of our institute, ICN National Research Institute. So I showed you this on purpose. So can you see any distinctness in this red flower circle? Just give a thought, no need to answer. Then, now I zoom it. Can you see something distinct is here? Now, if I further zoom it, what do you see? You see a white salvia among all the red salvia plants. So why it is so? Because if you see this other photo, you see a lot of red salvia for decoration, but what this single white flower is doing here, and why it is so? Because of mutation, because of some changes in the DNA that is responsible for producing the red color of this flower. So this is natural. Nobody has done any artificial thing to make this single plant as a white one, but this is a natural mutation. And this happens normally in nature. That's not abnormal. And nobody has genome edited to make it as a white one. Okay, so let me ask you, before going to the next slide, what do you think about agriculture and what you eat today? All the vegetables that you eat today, are they developed by natural process, or some human intervention, or some kind of artificial intervention is involved in that process? Do you consider agriculture as an artificial process or is a natural process? So agriculture, let me tell you the answer here. Agriculture is totally is not natural. Agriculture is not natural. Agriculture is an artificial process developed by human beings for their own purpose. Okay, so this is a photo of a wild corn plant, wild maize. So this is called teosinte, and it was 9,000 years ago. Our today's corn or maize, it looked like this. Only a single, this is a single cob. If you see, let me just show you. Single cob, and that has only 7 to 8 kernel. But what you eat today, this is our modern day corn. A single cob has thousands of grains in it, thousands of kernels in it. So this is because of artificial selection of human, because they realize that this is not sufficient for our food. So we have to select those teosinte plant that has a little bit more number of kernel. In the next generation, they selected for more number of kernel. In the next generation, they selected certain plants which have more number of kernels. And now we have this modern image which has huge number of kernel in a single cob. And if you have it, it is sufficient for your breakfast. So this is an artificial intervention. Now, if you see another perspective of agriculture, here, those are all teosinte plant growing in the wild, and you see how sparsely they are grown. They have, the distance between two plants are more. So if you want to grow 50 or 100 of teosinte plants, you need a huge area of land. Now, if you see the modern day cultivation practice, now you can grow the maize plant in certain way that 100 or 1,000 of maize plants can be grown in a very small area. So because this is actually help us to save our land, to save our resources, and to give us more power to harvest the yield in a very easy way. So we have artificially selected plants to increase their suitability to human needs. Like we selected for taste, we selected for yield, means which plant would give you more production. We selected those plants only, and then which helped us in storing our yields and also cultivation practices. So this is an example of cultivation practices, how we modified from this sparsely grown teosinte to a densely packed manner cornfield. So these are all agricultural practices, and these are all artificial things. Okay, so let me go further. These are all phenotypic example I gave you. Now go into the genome or some DNA level explanation. So this example, I think you have a lot of time seen it. So this is also a deliberate artificial selection for desirable traits. If you say this is a common wild master plant, and if you select them only for the stem trait, you will develop a kohlrabi. And then if you select for terminal bud, you develop a cabbage. Select for flower cluster, you develop a cauliflower. Similarly, broccoli, kale, Brussels sprouts, all are developed from a single Brassica oleracea because of human intervention and because of different kind of likings of different regions of the world. So these are all artificial selection. Now, yes, so this example of the left right panel also, you can see these are also examples of our own intervention to develop a more bigger size of fruit, to develop a more nutritious fruits. Okay, so in 1800s, mid 1800s, Gregor Mendel outlined the principles of heredity, and that was the beginning time of scientific plant breeding, because we are able to calculate, if you cross two plants, we are able to calculate how much of what kind of trait would be available in the next generation. So this is because of the Mendel's theory. Now, you see genetic variation. If I want to give you an example, the existing genetic variation in nature. So this is a natural genetic variation exists in rice varieties. So you can see a lot of different types of rice grains. And so a lot of variation. And this is because of why this kind of variation is visible from outside, from a naked eye, because of spontaneous mutation that happens in nature. As I started my slide with that white salvia among all the red salvia, because of a spontaneous mutation. This variation is also because of a spontaneous mutation. So up until 20th century, mankind could exploit the variation that are generated from spontaneous mutation, from natural mutation, and they selected those plants that are having some mutation and giving them a favorable characters because of that mutation. Okay, so this is the story of until 20th century.
Dr. Kutubuddin,
yes.
Just sorry to interrupt you. I'm not, just can you check whether your slide has a black strip on the top, or are we missing out something?
Oh, is it moving now?
No, it is moving. No, there is a black bar. Oh yeah, this is moving. It has brought it
because
down.
This is the bar of that Zoom,
Okay, so can you put it, okay, in the left, you can, yeah. I hope your slide is not getting disturbed for that. So you can put it in the left top. You can push that bar.
Sir, if you share with non-optimizing the share button, sir. When you are sharing, sir, you have to not use the optimized screen.
No, I have to share the optimized screen because there are a lot of videos. So that need to. Yeah.
So can you just take the bar and put it on the left corner
Yes.
top? Then
Left
that
top.
will be okay. Yeah, left top.
Yes, you can drag it extreme left
Drag
or
it.
right.
Yeah, yeah.
Okay.
You just drag it because you just now dragged it and you brought it at the bottom. Okay, this is better. This is better.
Okay. Okay.
Yeah.
Good. Thank you.
Okay.
Yeah, because these are maybe minute, but these are
No problem.
very important. These are very important. I understand.
Okay,
Yeah.
okay.
Because yeah, let me just, as it is now general discussion kind of thing. So if you listen to me very carefully, each slide to slide, you will get a lot of individual stories. And I think, the motto of learning is to keep something what you're listening. So I think you will have a lot of long-standing memory from my presentation if you be a little more attentive to each of the story that I'm going to share today. Okay, so now, okay, what I was thinking, I was telling that until 20th century, spontaneous mutation generated a lot of variation, and those variation are actually exploited by plant breeders or agriculturists to develop their crops. Okay, so now genetic variation, what we understood from this genetic variation is key to crop improvement. Okay, now when we learned that genetic variation is key to crop improvement, then we started pondering, then we started thinking that how to artificially generate genetic variation. Can we generate genetic variation artificially? Because spontaneous is very slow. If you go to the rate of the spontaneous mutations, it is 0.1 to nearby 50 per genomes per sexual generation. Means if one plant crosses one generation, maybe 0.1 to 50 mutations in the whole genome in a single generation. So it is very slow, and that cannot be exploited, and you cannot wait for that spontaneous mutation to happen, and that will give you a very good character. You do not know whether those spontaneous mutation would give you a good character or bad character. But because of the climate change and a lot of the other problems that we are facing today, we have to generate artificial genetic variation. So when people thought long back, then in 1920s and 1940s, they discovered X-rays, gamma rays, and alkylating agents, which can be applied to plants or any organisms, and that can randomly modify or change in DNA sequence. And when you randomly modify, this is an example of an X-ray, a gamma rays treated plants, and those are the flowers. Different kind of flowers are generated because there are random modifications, and those modifications resulted this kind of colors of the flower. Now, if I want to give you statistics, according to Food and Agriculture Organization, more than 2,500 plant varieties have been developed on the whole world by radiation-induced DNA modification. Now, keep in mind, radiation-induced DNA modification is a random process. You do not know in which part of the DNA is being modified. You cannot control that because the whole genome is prone to be modified. Okay, but we are having 2,500 plant varieties from that kind of mutation. Now, what if we have tools or methods that allow us to modify DNA at a desired spot? Because we developed in molecular biology a very great extent, and we know what gene plays what kind of role and what phenotype is controlled by what gene. And so do you have any kind of tools or methods that can very precisely modify this gene versus this gene? So precise genetic variation is on demand now. Now comes the CRISPR-Cas, which is the topic of my discussion today. It can be applied to modify DNA at a desired spot. You have learned a lot, I think, for the first few days. So this is the outline of my today's presentation. I will be talking about CRISPR-Cas9 mediated gene knockout. You already heard it, but I would give you a different perspective. In part two, I'd be briefly mentioning some other tools for gene regulation like CRISPR-A, CRISPR-I, and epigenome editing. Part three, I'd be taking a bit more time on precise editing, base editing, and prime editing. And part four is plant biotech innovation, some examples. Okay, so coming to the basic of my discussion today. So, eukaryotic organism like human being, as all of you, are made up of 37.2 trillion cells approximately. And also higher eukaryotic plants like rice have trillion of cells. Now, if you see in details in a plant cell, this is a plant cell and this is the nucleus. Now, this nucleus has chromosome, and this chromosome is nothing but a coil of long stretch of DNA. But the long stretch of DNA are not equally important. Every region of DNA are not equally important. Some portion of that DNA are functional in nature because they produce protein, and those functional portions are called gene. This is highlighted here in green color. And now, so gene is a functional unit of DNA. Now, why it is a functional unit? Because when the gene expresses, all of you know that a gene produces mRNA, and that mRNA produces a protein, and that protein ultimately governs a character. Okay, so what is there in our DNA? DNA has 4 different letters, A, G, C, and T. And the combination of that A, G, C, T letters makes a genetic code which ultimately almost governs everything in a living organism and a living cell. How do they behave? This depends on genetic code. Now, this is just short to introduce you. We all eat wheat, and wheat has 2 different protein. One is gliadin, another is gluten, and there's 2 makes gluten. And a lot of people, we eat wheat with very happy face, but some people, they are intolerant to gluten because they have gluten allergy. If they have it, they have a lot of stomach pain, so they cannot have it. Because of this gliadin protein actually produces an antigenic response or immune response. What is antigenic response? This body treat this gliadin protein as a foreign protein and is not good for our body. In some people, okay, and they start immune reaction against this protein. So they produce antibody, and they have this kind of destroyed villi, and they suffer from celiac disease. And because of gluten allergy, they have celiac disease, and they cannot absorb normally nutrition from this kind of wheat, rye, or kind of food. So now the question is, here is question for you. Is there a way to disrupt specifically the gliadin gene? As I told you, a protein is produced from a gene. So this gliadin must be producing from a gene, which is called gliadin gene. So can I specifically mutate or disrupt the gene? Which gene? Gliadin gene. So the answer is yes, because we have CRISPR-Cas9. And this CRISPR-Cas9 is called Caesar. This CRISPR-Cas9 can cause targeted inactivation of a functional unit of DNA. So it has 2 components, only 2 components. This is very easy. Forget all of the things that you learned for the time being. It is very easy. It has 2 components. One is a Cas9, another is called guide RNA. And if I want to give you an analogy, this Cas9 is, I call it as a postman, which has 2 scissors, okay? Cas9 is a DNA cutting protein. Analogically, it is a postman which has 2 scissors. Now, we have this is guide RNA, another part of this system. This guide RNA, I call it as an address. It's a genetic address. Now, you have a postman with 2 scissors, and you give this postman an address to deliver your letter, as you do. So, okay, if you give this address to the postman, then postman takes the address, and then it goes to the genome of your interest. Then it searches for the address. Where is present this address? Then it scans the whole genomic DNA, and to find the similarity with the address, what is this? This is an RNA. So RNA has this nucleotide, DNA has also the nucleotide. And whenever it says that this is the portion where I find a similarity with the address, then it binds there to the similar DNA sequence. And then whenever it successfully binds, then it sends or it gets a signal that this is the portion where I need to make a cut. Okay, now it has two scissors. So one scissor cut one strand of DNA, another scissor cuts another strand of DNA. And whenever your gene is broken by a double strand break, then the gene is non-functional. But how it becomes non-functional, that need to know. Okay, so how mutation occurs? We understood that double strand break happens, but how mutation occurs? So, you know, whenever there is a double strand break in DNA, the cell takes it very seriously. Why? Because DNA is very important for them. DNA is the hereditary material, you know, and as I told, DNA controls everything in an organism. How do they behave? So it is very serious damage to them. So they start repairing it. And there are two very prominent repairing system. One is NHEJ and another is HDR. But NHEJ is the predominant pathway, non-homologous end joining. It is nothing but to religate the broken end. Now, this NHEJ is predominant, obviously, but it has some tendency of creating some error or wrong thing during the repair system, Now, while repairing the two broken end, NHEJ inadvertently insert or delete some random DNA letters. Either delete some random DNA letters from the broken end or insert some DNA letter. So here I have shown in red color. Because of this random insertion or deletion happens in the gene sequence, the gene is knocked out. Why knocked out? Because, you know, as I told you, the sequence of this letter is very important. And the sequence is disrupted because of this new insertion or deletion. So the open reading frame of that gene became disrupted. So this gene would not produce the same protein as it was producing in its original form. So now your gene is not functional because it is not producing the same protein. Okay, so this CRISPR-Cas9 represents a wonderful tool to disrupt any gene function. Now, this HDR, I'm not going to into details, but this happens during meiosis, and this is very extremely low efficiency method. I'm not going to in details. So from this video, you'd be understanding in details.
Been adapted into a powerful tool for genomic research. There are two components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay, so I think you get what I wanted to understand. So now this is a DNA sequence, just to show you how this is a virtual DNA sequence. So you can see all the four DNA letters, they are arranged in a sequence, and that one gene sequence is different from other gene sequence, but the component is same. Component means the type of DNA letters are same, only the 4 types. Now, if you want to give or make a cut in this sequence, now you have to design a guide RNA. You have to give a address to the postman. So here is the guide RNA. You know, this is the target sequence, 20 base pair. You can select from the gene sequence, and that would actually act as your guide RNA or the address for the Cas9. Okay, so this is to just give you an example. Okay, now this is an example that I just introduced CRISPR-Cas9 with this example, the gluten allergy problem. And this has already been demonstrated that this is the gliadin gene region, and this is the region which is actually creates the problem of antigenic response in the allergic people. Okay, so what they have done, they made a cut here and here, and they knocked out the whole gliadin gene. And this is a fascinating data from that study, that you see this is the alpha gliadin in the control non-edited plant has high amount of alpha gliadin. The edited one has a very small and negligible amount of gliadin. So because of this specific disruption with CRISPR-Cas9, they could develop a low gluten wheat plant, and that could be eaten by the person which has who have gluten allergy. And all the other features like phenotypic characters are all similar to their counterpart. Okay, so now, as I told you, mentioned in my introductory slide, that when there is a cut, the repair mechanism in HEJ makes some random insertion or deletion during repair. It is a common notion that the insertion and deletion is random in nature, but while I was researching on that, and I could found that, while we are doing same experiment again and again, we are getting same type of mutation again and again. So why it is so? Then I pondered that why it is so. It should not be like that, because if it is random, so from this experiment, I got this kind of insertion deletion. Next experiment should give you a different kind of insertion and deletion. But this was not the case for me and one of my grad student, Justin. So then we thought that it has some pattern. It is not random. So then we thought that it could be predicted if we have a big repair data, means a lot of repair data, a lot of experimental data, and if you can feed on machine and develop some machine learning, then we could develop some models. That model would give you the idea that which guide RNA produces more mutation and what kind of mutation, means outcome they could predict. You can say, for example, if I want you to understand in a simple language, so this is a 20 base pair, your guide RNA, guide sequence, or the 20 base pair sequence in the genome. And if the -4 position from the NGG PAM is T, you have a single T, then if you make a cut with Cas9, you have a tendency of getting another T inserted. If you have a -4 position A, then it is likely that you will get mutation that another A is inserted. If you have double C at -4 and -5, you'd have a chance of getting one C deleted. So this is how prediction works. So let me give an example from our experiment. So these are all those guide RNA sequence, the 20 base pair sequence that I showed you. So and it depends on that fourth base, -4. That I showed you just in the earlier slide. All the -4 here, you can see the T. T is present at the -4 position. Okay, this is -1, 2, 3, and -4. Now, if you see, this is a chromatogram that we do during sequencing, and red color is T, black is G, and green is A, and blue is C. This is how we read the chromatogram in the genome sequence. So now, if you see here, when it did with ALS gene, we have seen another T inserted as expected, because the fourth one was T. Similar for another locus, PETA gene, the fourth one, this is the PETA, fourth nucleotide was T, and we got another T inserted here. So this is how our prediction worked according to the model. So now if you see, this is the percentage of frequency of one base pair insertion among all editing outcome. This is observed outcome, and these are the three models that we have tested with our data, and we have seen very close correlation with the observed and the predicted. These three are predicted, and this one is observed. Okay. Similarly, if you see the types of one base pair insertion, so in case of ALS, the observed, we have seen observed from our experiment is T inserted. In Delphi and forecast are the two models that predicted that T would be inserted. See, so it is very likely, but only a Sprout model did a misprediction. Similarly for PETA also, for CC also observed C, and in Delphi forecast predicted C. And this is also, you can see this is an interesting example of actin. Here observed, we have seen the largest insertion group is G, and the next insertion group is C. But in Delphi is a model, is a predictor model that predicted very precisely that the largest group would be G and the next largest group would be C. So this is very fascinating, and this is going to be published very soon. So now you have the power. So I'm going towards more precision of genome editing. It's not now random. You can predict your genome editing outcome if you want to even knock out your gene. Now, this part 2 has tools and techniques. Some tools and techniques I would highlight you. So now if you have a question, okay, I do not want to disrupt only single gene, I need to disrupt more than one gene. Can we do that with CRISPR-Cas9? Yes, we can do that. That is called multiplexing, because in one experiment, you can knock out or you can disrupt or you can edit more than one gene. How? You have the same postman, you have to give different address. Very simple. Because suppose you posted 3 letters, your marriage invitation card, to your 50 friends, and you only read what was the difference? Same postman, card is same, only the address is different. Okay, similar here, your guide RNA should be designed or specifically for each gene you want to target, and the same postman would take each of the address and they will deliver, means they will go to the genome and they will make cut at locus 1, locus 2, locus 3. It means gene 1, gene 2, gene 3, wherever you designated by the guide RNA. Okay, so harnessing this one, let me give my example of my work here.
Hello? Hello? Sorry. Hello?
Yeah. Any problem?
Yes. What is it? Hello?
Is there any question from Manju Rani?
Question? Manju, do you have any question? Okay, maybe
Okay,
we can take, we can take the questions towards the end. So all of you kindly keep your questions ready with you. Let Dr. Kutubuddin proceed with his talk, and please keep the questions ready with you. We will address all of them, so don't worry. Okay, Dr. Kutubuddin, you can proceed. Hello?
Sure. Yeah, right.
Yeah, please, you can go ahead.
Okay, so this is a fascinating work going on in our institute, just from our group, so let me share it to you. So you see 2 different rice fields in the same slide. This one is, what is it? And this one, what is it? Both are rice, but this is standing rice and this has fallen. Because this is called a lodging problem, because if you have high wind or rain, you have this problem. And if it happens in rice field, then farmers are very, very sad because they cannot harvest properly and they lose a lot of yield. So this is a real problem, and that was a problem long ago. All of our grandfather probably knew that, because the rice plant was very tall and it could not, withstand with a long heavy wind or heavy rain, and that's why they lose a lot of the production that they were expecting. Okay, but this problem has been sorted out during Green Revolution, because we developed shorter rice plant and that have long, is very strengthful, the stem of rice, so it can withstand the pressure of wind and rain. So now that was the story of Green Revolution. Now, if you see, so we want to mimic the Green Revolution by genome editing, by developing semi-dwarfing economically important land races. This is an example of that Green Revolution that I just told you. This is the kind of rice we used to have earlier, very long. And when this is a very small dwarf variety, when both of them are crossed, and this Green Revolution rice has been developed. The first Green Revolution rice is IR8. Okay, and that has actually revolutionized, as it named, green revolution, revolutionized our agriculture, and we get to be self-sufficient in food production because of this development, okay, because of reducing the height. Okay, now what happened to that when it was analyzed in detail to why the height is reduced? Because, you know, there is a gibberellic acid, or gibberellin is a plant hormone, most of you know probably. So this gibberellin actually responsible for plant height, and when they have analyzed in this semi-dwarf cultivar IR8, they have seen that one of the biosynthetic gene, this is called SD1, has a 383 base pair deletion in that gene. I mean, some portion of that gene is deleted. Where? In those semi-dwarf or those dwarf cultivars or dwarf varieties. Okay. Now, so because of this mutation only, it was very helpful for human being to get green revolution. Okay, now this kind of development actually gives us better lodging resistance, adaptation to high nitrogen fertilizer condition, gaining high harvest index, and also many other features. So what we wanted to do, because it takes, you know, if you cross the two variety, one long and one small, then the whole genome being shuffled. So you cannot specifically transfer this deleted gene to your target variety. And it takes 7 to 8 or 9 generations to get a pure variety, okay? But still you have a lot of residual material from this long variety, and which is detrimental, and that is called linkage drag. Now, if we have genome editing tool, so what we have thought that what if I make a cut here and make another cut here by giving 2 address to Cas9, can we mimic a green revolution allele like this one, okay? So this was our hypothesis by making this 2 cut. Now, so we failed a lot in the beginning, but these 3 dedicated students, Devashmita Monashini and this is my friend Shubhashish, who was working as an RA. So they are a very involved person in this project. So, you know, so we designed 2 guide RNA and we made double cut, and that was expected to delete a 621 base pair according to our analysis from software and from computer sequence analysis. Okay, now when we did a real experiment, and we have seen here, you can see this is a control one. When did a PCR of that gene, it amplified 1300 base pair as a fragment. Okay. So 1300 base pair is the control one, and we have seen some plants, they give rise a smaller band with the same primer. It means the in between base pair band, what does it mean? It means 620 base pair deleted from the 1300 original one. So some plant we got deleted, some plant we have not. That is expected. Okay. So when I did it, so if I want to show you in detail, so this is a chromatogram from the wild type or control one, the long plant, and in between there was 620 base pair. I cannot show here because it's a limited slide. And this is the mutant that we observed recently, that this portion and this portion is ligated, and in between 620 base pair is deleted. So this is wonderful that we got, and we're very excited. So it is going to give rise, because this is the one that is unedited. This is a long lodging susceptible one, and when we applied CRISPR-Cas9, it is expected to give rise a semi-dwarf on a small dwarf plant that would be lodging tolerant and increased harvest index. Okay, so that excitement apart, now I'm going beyond that double strand break in the genome. So can you do something different with that? So I have to go fast. So, you know, this one I already described. Now, what if you snatch one scissor from the postman, and if you do a protein engineering kind of thing, single mutation in this Cas9 gene, this Cas9 become a Nikis Cas9, so that it has the ability to make a single strand cut, not a double strand cut. This is called Nikis Cas9. Remember. Now, this is another thing. If you do double mutation in this Cas9 gene, then both the scissor is gone. So it has the ability to bind, but not cut, not making any cut. So dead Cas9 can bind to a specific DNA sequence as per the address you are giving, as per the guide RNA you are giving, but it cannot make a cut there. So you learned here two different things. One is Nikis Cas9, another is Cas9. Okay, now, so scientists have harnessed it for playing with the gene regulation. You know, Cas9, as I told you, Cas9 has the ability to bind to a certain region. So if you give them a guide RNA which is targeted to enhancer or promoter, and Cas9 would go there and they will remain bind there because they don't have the ability to cut. But what if you fuse another protein with the Cas9? Now, if you give something with the postman, okay, and you tell postman, so okay, supplying with go to that place and supply this protein there, okay. So Cas9 can supply that specific position, okay. So here CRAB domain is fused with Cas9, and this CRAB is a transcription repressor protein. So once you give a guide RNA, Cas9 fused with CRAB will go to the enhancer or promoter. It will remain bind there, and because of the presence of Cas9 and CRAB there, the transcription of that gene would be inhibited. Okay, so this is called CRISPR-I, CRISPR interference. Now similarly, CRISPR activation is there, where the repressor domain is replaced with an activation domain. So Cas9 with guide RNA goes to the promoter and takes the activator protein with them. Now, because of the presence of activator protein in the promoter sequence, then your gene expression would be higher than in the normal. So suppose you do not want to edit your gene, but you want to express higher your gene, you can use this CRISPR activation tool. Now, similarly, you know, in some cases, you see the same gene is present in two species or two genotypes or two varieties, but the same gene is not expressed in a similar level. Then what happens? Because of their promoter methylation, if the promoter is methylated, then the gene expression is inhibited normally. Okay. Now, do you have any answer to remove this kind of epigenetic signature? This is called epigenetic signature. Gene sequence is same, but the modification is different, and that is why expression is different. Okay, so Cas9 has been harnessed similarly. Cas9 is fused with TAT1. TAT1 can demethylate. Okay, so when you give a Cas9 TAT1 fusion with a guide RNA targeted to a promoter that is already methylated. So it goes there, and you see this blue bubble is the methylated bases. Now, when Cas9 and TAT1 goes there, TAT1 do the demethylation work. And when it is demethylated, then the gene is expressed in higher amount. So this is how you can modify even the epigenome of your target organism. Okay, so now coming to the last, not last part, I have fourth part. So this part 3, precise editing. So the question here I want to put in front of you, I think all of you know that this is Oro and this is Oro's friend. So Oro is 12 years old. His friend is also 12 years old, but Oro looks like 83 years old. Why? Because Oro is suffering from a deadly disease that's called progeria. That causes rapid childhood aging, and it looks like very aged person. And they die within 13 and 14 years of their age. You know why? Because of a single difference in their genome. What difference? There is a gene, and in that gene, the healthy child has G, and Oro had A. And this single wrong letter caused Oro to suffer, and Oro's family to suffer, Oro's relative to suffer like anything. We knew this reason, but we did not have any answer to cure. Now, another example, if I give you on plant perspective, this is the same plant, Kitake. Now, if this plant has a C letter in a gene, BSRK1, it is susceptible to a disease, fungal disease. Now, if it has a T instead of C, it will be resistant. So this kind of single wrong letter can do a lot in governing many important phenotypes. Now, this is my question for you. Is there a way to rewrite genetic letters precisely? not the way to disrupt a gene. You have to rewrite precisely. You have to do G to A, A to G. You have to do C to T here. Now the answer is base editing, because base editing is kind of pencil. So this pencil has, in one end it has a rubber and another eraser, and another end it has a, the lid. So you erase the wrong letter and you insert the right letter. So that's why I called it as a pencil base editing. And we have this field has been emerged like anything, very rapidly expanded field, and we have the ability now to convert A to G, G to A, T to C, C to T, and C to G, G to C. These are the changes that we could install in our genome with base editing. So what is it? In base editing, we use a Cas9, Nikis Cas9, not the normal Cas9. Nikis Cas9 matlab single scissor, and it is fused with a deaminase. This deaminase actually do the work of modifying the target nucleotide, and this has been developed by David Liu from MIT. And yeah, this is the Nikis Cas9 that has been used in base editing tool. I will be discussing a lot later. Okay, so let me give you a simple example here. What do you see here? Some mistake, twinkle twinkle little. This is the mistake, okay? And there is no star in the sky because it is not expressed because of this wrong letter. Now, base editing is a tool that could correct this letter in the DNA, and you get corrected, and you get expression of your gene, and you get a lot of star. Okay, so this is how base editing works in the genome. I'm not going to say anything more than that. I think all of you could get my point. Now, this is the small, very brief video.
And then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay, so this is the first type of base editor that I was talking about. One is cytosine base editor that can cause C to T in one strand, in another strand. You can see if this is the base pair CG, it could convert TA. Okay, so what is that? This is Cas9 fused with a cytidine deaminase. This is a cytidine deaminase. This is also another cytidine deaminase. And with that, UGI, uracil glycosylase inhibitor, is another protein is fused and developed this base editing tool called cytosine base editor. Okay, so it can install C to T mutation. Now, the second class is adenine base editor that can cause A to G. It means AT base pair would be converted to GC base pair. And it has simple structural details in Cas9 fused with deaminase, that is the adenosine deaminase. Okay. Now, how this adenine base editor works? Because suppose you have an AT base pair in a defective gene, and you want to correct it to a GC base pair, as it was the case of Oro. So A was the wrong base and G was the correct. So you want to do this kind of correction. What do you do? So you apply ABE, adenine base editor, and as I told you, ABE has adenosine deaminase enzyme fused with it. So this adenosine deaminase will deaminate adenine here, and it will make an inosine. And now you see I paired with a T, and that's a mismatch. It should not be like that, because T always should pair with A. Okay, so I paired with T. Now cellular mismatch repair mechanism, they understand this mistake or this mismatch, and they want to remove one of the mismatched base pair. This is the common mechanism. Now, which one they will remove? I is mismatch or T is mismatch? They will be confused. Okay, so there is a very intelligent and clever designing of the base editing tool. So they did Cas9 so that single strand break is made in this T containing strand. So when T containing strand has a break, then cell will understand this strand is wrong strand, or this strand is damaged strand. So the T should be removed. Okay, when the T is removed, then opposite of I, there is a correct base. I always pair with C. So IC base pair is formed, and in the next round of repair and replication, it becomes a GC, or you get corrected gene. So this is how adenine base editor works in the cell. Now, what we did, we tried to target both two loci at once by using this base editing tool in RISE. And we have done this kind of multiplexing approach where we fused a tRNA sequence with a guide RNA, another tRNA, another guide RNA. So this is a complete polycystonic tRNA gRNA. So what it actually gives rise, why we have fused tRNA? Because once it goes into the plant, so the tRNA is cleaved automatically by RNSP and RNSZ, and your guide RNA becomes separated. So you do not have to deliver them separately. You deliver them together in the cell, which is easier. And then in the cell, tRNA would be cleaved and both the guide RNA would be separated. And the WSL5 would go to chromosome 4. This guide would be go to chromosome 4. And this guide would go to chromosome 3 to their target. Okay. Now, when we applied, we have some very exciting result. Just see this one. This one is our target chromatogram of the gene. This is a small region I have just highlighted here, and we targeted this A base to change it. Okay, and when we treated with base editor, we got precisely changed A to G. You see the green peak, which actually indicates A nucleotide has been converted to a G nucleotide very precisely. And if you match the other color of the two peaks, you can see all others are similar. Only this one, targeted nucleotide, has been changed by adenine base editor. And we knew why we have selected this one, because this WSL5 means white striped leaf phenotype. So when we did this kind of SNP mutation or single nucleotide or single letter change, we got this kind of expected mutant phenotype, white striped leaf, because of this single change. Similarly, for our second gene, that's called Zebra3 or Z3 phenotype. So this is the chromatogram where we targeted this A letter, and we have got similarly modified G letter by applying base editor, and all others remain intact. Only the target one has been changed. And as a result, we have got the phenotype also. This is the light green, dark green, light green, dark green, light green, dark green phenotype, as it is named Zebra3, alternative patches of light green, dark green. So this is, you already got the point that single nucleotide is very important for many reasons, and we can now we have the power to install or correct this kind of mutation by base editing. Okay, so now with this technology, CRISPR-Cas9 has a very beautiful aspect that if you apply this technology, you can develop a plant or mutant of your interest without any residual effect of these kind of construct or the DNA that we are transferring into the plant. So you can develop non-transgenic one. So this is an example from our results. So, this is simple PCR to amplify the base editor construct. And if the band is present, it means base editor is still present. If band is absent, base editor is gone. Okay, so these are all mutant. We selected and we did a PCR, and we have obtained out of 25, 6 plants are mutant but did not have any base editor present in the genome. It means they are non-transgenic. But how it happens? Let me explain a little bit. So here is base editor. Suppose base editor, when you transform, it is supposed integrated into chromosome 1, and your target is in chromosome 4. Now, base editor produces a protein and it goes to the target and it modifies the target. Okay. Now, once the target is modified or target is mutated, okay. So let me know, what do you think? Do you need the base editor to be present in the cell still? No, because your target is modified. So the work of base editor is done. You do not need the base editor to keep in the genome. So in the next generation, by virtue of sexual segregation, you can select those plants that have only target mutated but does not have base editor. And you get a homozygous mutant without any transgene integrated, as in the case of this kind of absent of band. This simple PCR you have to do, and you get mutant plant without the base editor integrated. So you get non-transgenic plant very rapidly with this method. Okay, so now we moved forward to apply this amazing technology to develop something relevant to farmers, something relevant to us. So, you know, in a genome, if you see this kind of sequence, codon TAG, it's a stop codon. It means this is a stop signal to the translating machinery of the cell. Translating machinery, when the CTAG is present, it stops there. So it's a stop signal, okay? So in many genes, this kind of stop signal, normally this kind of stop signal should be present at the end of a gene, okay? But in many genes, due to some mutation, natural unfavorable mutation, this kind of stop codon may be present at the early position of the gene or at the mid of the gene, but not at the end of the gene. So what would happen? The gene would not be translated, and gene would not be producing the long length of protein that it requires to produce. So the gene would be producing a because of the premature stop codon, it would be producing a truncated, a small version of protein. And because of this kind of thing in one gene, rice plant become disease susceptible. Okay. Now we thought that we have adenine base editor. Can we just convert this A to G? So we tried. Yes, we are successful to develop TAG convert to TGG, and this stop signal is gone. Now the protein synthesizing machinery get a green signal here, and they would force it further till the end of the gene, and they will produce a long length of protein, a normal protein. And because of that, this plant become disease resistant. Okay, so this is we already achieved, and this work is also going on. So this is how you can apply this for crop improvement and many other purposes. Okay, the last almost at the end of my presentation, this is another precise editing thing that's called prime editing. What do you see here? Sickle cell, normal RBC, red blood corpuscle. Now, you know why it happens. In many cases, there is a A2T mutation in the beta globin gene, and this single wrong letter causes sickle cell anemia. Similar, Tay-Sachs disease. Because of a 4 base pair insertion, TATC, in hexagene. These are all normal. Nobody did. This is normal. Natural mutation occurred in human body. TATC insertion in hexagene causes this disease. This is a deadly disease, and child dies within age 5 if you have a Tay-Sachs disease. Now, do you have any tool to correct with the available tool that I discussed? Yes, we have the tool to disrupt gene. We have the tool to correct single point mutation. But no, I did not say you A to T. You cannot do A to T with base editing. We do not have that tool yet. We have A to G, but we do not have a tool to specifically delete 4 base pair. Here comes prime editing, where you can do all 12 types of base substitution, including transition and transversal mutation. You can insert specifically what kind of insertion you want. Up to 44 base pair you can achieve. Deletion also up to 80 base pair you can achieve. A specific deletion, not random deletion. Combination of all above 3 can also be achieved with prime editing. And this has been also developed by David Liu's group, by Angelo. So, you know, here also Cas9 has been used, but the Cas9 is fused with a reverse transcriptase enzyme. Cas9 is fused with a reverse transcriptase enzyme. Not with deaminase. Remember, not with deaminase. For base editing, we fused Cas9 with deaminase. For prime editing, we fused Cas9 with reverse transcriptase enzyme. And another modification is installed. This is a normal guide RNA. You know, it looks like this, but in case of prime editing, we have to use a prime editing guide RNA, where the 3' end here, 3' end has some extension, and this is called binding region and edited sequences, and it looks like this when it binds with the genome sequence. Okay, so now I'm not going into details with the mechanism of this prime editing technology, but you can read more. I will give you the resources. So this is the last part. Only few slides I have left with me. So I will just give you a glimpse because I cannot present everything what is going on in plant field within 1 hour. So we recently wrote this review article about base editing and prime editing in plants. If you are interested, I can send it to you if it is not available to you, and you can get a lot of different ideas of what kind of application could be done. So this one I have not mentioned, but there is a recent base editor development where you can also modify mitochondrial or even chloroplast genome, not only nuclear genome. I talked in my earlier slides about nuclear genome editing, but you know, mitochondria and plastid also have their own genome, also have their own DNA, and that could also be edited with this organelle base editor recently developed, so that you have a lot of different power to modify chloroplast genome for developing crop, for improving crops. Okay, so you know, those base editing, prime editing has been used in many plants. These are the representative image for herbicide tolerance, nutrition, altered nutrients in high sugar content in strawberry, increased carotenoid in tomato, altered starch composition in rice, and also it has also been applied in rapid domestication. You know, as I told you, from teosinte to corn, it took how many years? 9,000 years. Now, from wild rice to cultivated rice, you could apply this tool to do same kind of thing in 4 to 5 years, in 2 to 4 years. So, you know, you have this power to rapidly domesticate your wild crops. And obviously, it is very much useful for yield improvement, as I showed you one example from my case. And disease resistance, also I showed you an example of correcting a stop codon for developing disease resistance. There are a lot of other ways to do it. Input use efficiency, you know, this kind of fertilizer use is damaging our environment. So if you can develop a crop which has better input use efficiency, better fertilizer use efficiency, so that you apply less fertilizer and you get more production. Okay, that could be also done with this. And you know, many new traits that we never thought of is also being developed with genome editing. Rapid customization of Solanaceae fruit crops for urban agriculture, that has been recently developed from Jackery Lippmann's lab from Cold Spring Herbal Laboratory. So, so this is a snapshot. You know, a lot of different agricultural crops have been already demonstrated to be editable with these tools for different kind of desirable traits. So, yes, last slide. The question is, yes, everything is in lab, or is there any crops that are commercially cultivated? This is the genome edited soybean, already grows in USA field. And this soybean is called Calino, which has high oleic acid, and it has the property like olive oil. So you get soybean oil like olive oil, healthy benefit like olive oil. So it is in USA growing, and this is the Professor Dan Hoytes, who is the developer of this one. So this, and another one recently commercialized in Japan. It is being sold. You can even try to buy it from online and import it from Japan. Japan breeders already launched it in you know, this is 11/1. 2021. So they already started selling it. This is called high GABA amino acid, and it has an amino acid called gamma aminobutyric acid. And if the tomato has more amount of this one, if you eat that tomato, it probably helps you to relax and lower your blood pressure and so lower the hypertension. This is called high GABA tomato. So these two crops already commercialized in a very rapid time when the genome editing developed. 2013 first paper, 2012 first paper came, and this is rapidly expanded, rapidly applied, and rapidly commercialized. So a lot of other crops are in pipelines. Okay, so if you want to know more, so these two Springer protocols, both protocol is to help others who are new to this. And so we thought of this one during a meeting in the USA, and me and another, my co-editor, we discussed, and we came up with these two books, which has a lot of protocols to apply in different organisms, like in fungus, in plant, in bacteria, and how to apply this technology. There is a stepwise protocol and material methods, what materials are needed, okay? And for easy understanding, you should go through all the reviews. That's why I listed here. So these 3 reviews you could go through to understand their technical details and their application horizon. Okay, so if you need any paper, just write it to me. I would send it to you. Even I would be happy to share a bunch of those PDF with Dr. Jaya, and she can share with all of you guys. Okay, so yeah. Thank you, and I must acknowledge the funding from all those agencies: NRI, my institute, ICAR, USIF for Fulbright, and USDA, NSF, and these are my lab members and my supervisor of my Fulbright tenure, and these are the group that I'm working with now in NRI. Okay, thank you very much for giving me this chance to talk in this FDP program.
Wow, thank you, Dr. Kutubuddin. And it was a fascinating lecture. As you said, that the stories that you had to share, that was really, really wonderful. I think all of them have been captivated with your lecture. I have questions, but first let us open the questions for our participants. Yes,
Sure.
There were some hands raised earlier. And Vignesh, you can unmute and ask your question.
Yes, ma'am. Good morning, sir.
Good morning.
Sir, your talk was really good, sir. I understood from the basic concepts that made me to think over on the advanced issues onto it. So it might be looking too even basic, but the doubt what I had was when you were giving a talk on the 680 base pair removal from a particular gene.
Yeah.
So what I was looking into is that even a small change in the genome, a small change, it might lead into a drastic change in the future. So when I'm gene editing a particular crop and I'm removing a particular segment out of it, and in the when this crop is coming into the commercial purposes, and if it is by mistake given a crossover with the wild breed, is there any chance of any drastic change or effect in the genome?
Very good question, Vignesh. I got your point. So yeah, so let me show you. Just think. Okay, that's a good question, and good. I think you should ask this kind of question. So that's not silly question. That's a very important question. Okay, let me show you. This is the green revolution. I gave you example. Okay, so this deletion is naturally existing deletion. And the whole world is eating those kind of rice plant, and they are breeding from this rice. So we're transferring this rice for transferring the deletion trait because it is beneficial for us. Now the same thing, if we could try to do it in the landrace that I'm trying to do, it took a lot of time and a lot of negative impact could be transferred from this gene, because I want to transfer this deleted gene. This is my target. But this is called linkage drag, from this gene, other gene can also be transferred, which are negatively impacting gene through traditional breeding. Now, what I am doing with gene editing, I am targeting to delete a similar fragment from the same gene. Same genes means the gene that does not have deletion, I'm causing a deletion to make them short. And now if you ask me, all the plant that we eat now, rice, all are semi-dwarf and all has 383 base pair deletion. There is no problem, no detection. And farmers and breeders are breeding it from long time, from 1960s. So as it is already proved as safe and it is beneficial, we are mimicking. Here you can see my title, Green Revolution Mimic. I'm not doing any new kind of things. Okay, have you got my point, Vignesh?
Yes, sir. So it's always good to like understand the concept and mimicking what is existing.
Yes, you're right, exactly. So it is already, the deletion is already present in wherever you go. If you amplify this SD1, you would get 383 base pair deletion existing in the field in front of your house. Same thing, we are trying to do it because we need to do it rapidly, and we do not want to incorporate some undesired gene with this one. That is fine. Okay.
Yes, sir. Thanks a lot, sir.
Yeah. Now, any other?
Sir, good morning, sir.
Good morning.
So very nice talk we had. So just have few questions. One was like initially you talk about that prediction tool. So it was predicting based on like what guide RNA and at what base pair you have, and that you can actually predict what change will be there. So that means, is there any relationship between that and the PAM sequence, or we're ignoring the PAM and just focusing on the sequence of the guide RNA only? Because if we are using the PAM, you're restricted with the guide RNA sequence. We can't.
No, yeah, you're right. So we have to consider the PAM, because I have not, I just deleted the slide before my presentation, because I know PAM is discussed a lot last few days. To make my presentation short, but PAM is obviously should be considered during prediction. So your guide RNA sequence and then PAM all should be considered during prediction. Yes.
Okay, sir. Thank you. And one more question, like using this CRISPR-Cas9, and what we can do in the medicinal plants, what kind of evolution or what kind of studies, what kind of what we can explore in the medicinal plants, because
yes, medicinal plant is an amazing field that could also be benefited from this field, because a lot of people are doing this to increase the active constituent of your medicinal plant species. Suppose it has suppose curcumin. Okay, so curcumin, as I showed you, that high GABA tomato. So it is a kind of medicinal plant. Obviously, it's a vegetable, but it's now high GABA tomato. Why people buy it? Because it has a certain constituent in a high amount, which would give you to control your blood pressure. Okay, so that's the medicinal property could be increased using CRISPR-Cas. In many other plants also, I know that there are, the medicinal constituent biosynthetic pathway. So they are modifying the biosynthetic pathway to modulate the expression of the active constituent.
Okay, sir. Thank you so much.
Okay, Anuj.
Am I audible, ma'am?
Yes, you are.
Yeah. Good morning, sir. And it's such an excellent talk. And actually, I have a question that in the last 2 or 3 slides, you are mentioning about the commercially cultivated plants. So I want to know, is there any such plant that is being cultivated in India right now?
Oh, no, not genome editing, because we are in the process. We do not have a regulation yet. So government is framing a regulation how to regulate genome edited crops. So DBT, Department of Biotechnology, is working on that. They have made a draft regulation, and they already indicated on requested government to deregulate genome edited crops from the purview of normal GMOs. Genetically modified crops will regulate a lot, but genome edited crops is not going to be regulated if there is few changes in the genome sequence. But we do not have a regulation, so there is no question of any commercial cultivation. Okay, but a lot of Siddharth Theory told about banana probably yesterday. So his crop in pipeline. So once the regulation is done, or regulation decision is taken by government of India, his crop would be in the market. And also from Viswanathan Chinnuswamy from IARI, you have developed a drought resistant rice that can grow in low water situation, limited water situation. So there are a lot of other people also developing a lot of other traits, but they're all in the lab and in the greenhouse, but not in the field till now. Yeah.
Okay. And second question, as you are just telling now that you are just mimicking the natural deletions in the editing process. And so what about that? Because most of the genes are multifactorial and there is not one gene that is regulating a particular function. There might be some functions which are regulated by multiple genes and you are just knocking out one gene or mimicking one gene. Does this have deleterious effects or what effects you are expecting from that editing?
Yeah, this is kind of similar question what Bigness has asked me. So, we know from last 40 years that this SD1 deletion, this is a single allele governed character that is tall and dwarf in case of rice. So we know from 50 years of study of molecular biology and genetics. That's why we are confident on it. And that's why we go on developing this kind of plant by deleting. Okay. So we have a lot of background study already in the literature, huge amount, 50 years of study. You see, same green revolution has been also applied for wheat. Wheat was very long, and there was a gene RHT, reduced height gene. Same kind of non-functional gene was there, and they are also trying to mimic that one. So there is no way that it should create other problem. But obviously, before going commercialization, there are a lot of process to evaluate. If your plant, suppose you develop 1,000 plants, if your 1,000 plant, maybe some 5 plants had some deleterious effect, you discard those 5 plants. Now, if you have very pure plants which does not have any other impact, the quality of rice, the everything, cooking quality, eating quality, height, all other things, all are perfect compared with the control one. And if it performs equally, then we go for commercialization. So there is a long process. So it's not that developed and sent to farmers. It's not like that. It's highly regulated.
Okay. And sir, what if we are going to do this kind of studies in some other plant and there is a multifactorial, then we have to look out
Yes,
for.
whenever you select a gene, from the literature, because this is a research. Means when you select a target, we did a lot of ground study. If we edit this one, what would going to happen? Because this gene has this kind of multiple effects, should not target this one. Yes, you can target this one for study, for biological study. Okay, disrupting this gene, what kind of effect is going on? But for not developing a product. Developing a product means you go for that gene. That gene is very highly studied. You have enough literature to support that the deletion would cause the desired phenotype and would not cause any undesired phenotype.
Right. Dr. Manoj Kumar?
Okay, thank you so much.
Yeah.
Thank you so much.
Am I audible?
Yes.
Yeah. Hi. Hi, Dr. Qutbuddin. Nice meeting you. And I think we are meeting first time, but I have seen your work. So I am Manoj, and I have been working in genome editing for last 7-8 years. Yet to publish a single paper, but my papers you can see in Science, etc. So I worked with Phil Wig at Cambridge University, and I did my PhD from Max Planck Cologne. So because I have been kept quiet because I know Devajit and etc. So I have been listening throughout the day, ma'am, but I was not interacting much because I didn't want to interfere. All are experts. Here there are few things I want to add. That first of all, because maybe majority of people probably are not from plant background, and I am a cell biologist and developmental biologist, although I work on plant and basically my work is on cell cycle. So few things I would like to say that agriculture is not artificial. You started with this. It is a very natural process. Yes, farming is artificial. That I agree, that we select trait. This is very important. It is agriculture is completely natural, and we select certain trait, and that we call farming. Like we choose and second, the picture you showed on the flower thing, you said it is mutation. It is not mutation actually, that plant. It is RNA interference. And RNA interference is not because of mutation. So this is for the people, attendees, that yes, mutation is there. We know APA status. We have different phenomena. And genome editing is in principle many things. I mean, you have been doing on This gliadin, I wish you all the best, because I also work on it and I find it extremely difficult, because to maintain the texture after mutating gliadin is getting too much challenging. There are, I think, 3-4 papers who have developed gliadin mutated wheat, and it's very difficult. And a few other things that we cannot really order anything genome edited in India at the ethical end, whether it is in US or whether it is in Japan. We cannot order it. We cannot bring any genome edited crop in India as per ethical regulations, because you might know that just very adjacent to our country, Bangladesh, it has allowed brinjals, even transgenic brinjals are there. So on that note, for the normal, if I may say so for the attendant, in plant, it is very easy to do things because of regulatory aspects, because not many authorities are not, that much surveillance is being done, but it is extremely difficult to do at the bureaucracy level. So we cannot just do it. And we cannot really get all these, although we know that there is a debate, whether it is by Michael Weven from John Innes or all these big people, but it is still considered as a transgenic plant. EU has not approved it, right? So we have to be careful about all the aspects, and because public is public. So when we say something, they just believe. It doesn't matter what is the background. So I request you, Maulana. So, and I know many of your colleagues, I mean, Pathak sir was there. We know very well each other, and yeah, but nonetheless, you are publishing excellent. Highly appreciate your work. Nature Plant Review is a very nice review. And the earlier review, CRISPR-Cas9 method, I think it's in plant biotechnology. That's also very well written and very explanatory. Yeah, so these are my few points for.
Okay.
Yeah, you may please correct me if I'm wrong.
Okay, so let me just come one by one. When I'm introducing any subject to audience, that's why I asked Dr. Joya previously, who are the audiences? Introducing a subject, agriculture and farming differences, I want to just tell them that farming, how we interfered, it's not all natural. As I told you, artificial selection example. So I need not to be scientifically super correct when I'm introducing something. 1 second. About the second one is RNAi example of this one. That could be because of RNA. We do not know. We have not analyzed that white salvia and red salvia.
As well. Yeah.
1 second, but can also be done by some mutation. Some pigment producing gene is mutated. Who knows? That's why this is an example. It's not I have analyzed and I said this is because of some mutation already there. It could be because of RNA, and that RNA is also an interference. There's a second point. Third portion is difficult. Every experiment is difficult. I showed you in one example that SD1 deleted 6 ATPase pair. We struggled for 2 years and we got success now only because we used other guides that did not work, and then we designed. Second time did not work. Third time it now worked, and that I showed it to you and to all the audiences. So, but I cannot show all my failures in 1 hour. Okay, it's not a long interaction. Okay, so this is one thing. Difficult is difficult, but what scientists show, their success. Another thing is UK. What you said, UK is highly criticized all over the world because of the UA Court of Justice regulatory comment on genome edited crops. Now you must be aware about that. No, that's for European Union, but UK is going to allow field trial for wheat, low asparagine wheat. You just, if you Google it, you can get it. Sweden already, Spain, Belgium, all are going positively towards genome edited crops. Okay, so European Union probably turning positive towards this one. Okay, and what you said, I also echo your voice. That what scientists say, public is public and public believe what we say. And that's the problem with genome edited crops and genome modification, So a lot of things, a lot of people, they show some derogatory comment, which is non-scientific. just to show or spread fear monger, and public believe that. So that's the problem, especially problem of science communication to public, because there are a lot of people who spread non-scientific nonsense, and people believe that because people does not have time to read on all these things, and it is easy to spread a fear news than a good news. So that's my purpose of saying, and thank you for your comment.
Thank you.
Yes, thank you.
Yeah,
Thank you,
well
Dr.
addressed.
Manoj. Yeah.
Thank you.
Dr. Umesh, if you can unmute and ask your question. A quick 1 or 2 questions we can take. Dr. Umesh? Okay. Meanwhile, I just have one.
Hello.
Okay.
Hello.
Please go ahead.
Madam, good morning. Sir, good morning.
Yeah.
Good morning.
Okay.
This is Ravi Prasad from Odisha. Sir, your talk is really excellent, so informative. I've been hearing, and I was having a little bit confusion in the CRISPR-Cas9 technology, but today it is really very fundamental. It is really expressed. They are expressed in such a way that is fundamentally very clear. But I have a question, sir. The question means I have a doubt in that. Whatever product produced, organisms developed through CRISPR-Cas9 technology, can we call it as a GMO?
Yeah, that's a very good question. So some country call it GMO, that as Dr. Manoj Kumar said, European Union call it as a GMO. Why? You know, the reason they have given is very funny reason they have given. They said it is not natural because human is intervening. And they said mutation breeding is natural because mutation breeding is not natural. But as we have proved that mutation breeding does not have any impact, because we have history of cultivating safely the mutation breeding. You know, mutation breeding, I give you example. You expose the whole plant with gamma rays, okay, radioactive rays, gamma rays, and the whole genome is mutated. And then you generate 1,000 plants. After 1,000 plants screening, you get one desired result, and you isolate that plant, you grow the seed, and you grow next generation. This is the type of mutation breeding. But European Union said that mutation breeding is okay because it is human intervention, but we have safe history. Genome editing human is doing, and we do not have safe history. That's why you call it as a genome modification, means GMO. That is the view of European Union. Now UK is out of European Union, and other countries, like I said, Sweden, all are going very positively to allow field trial of genome edited crops. This is of Europe. Now if you see, Japan already approved non-GMO, no genome modification. They should not be labeled as GMOs. They are normal mutant breeding, okay? And now USA already approved, and USA is going to approve a lot of other things. Low, waxy corn is also in the pipeline. So, and Australia also in the mid ground. China probably going to approve it as a non-GMO. So whole world has a positive vibe, obviously, about genome editing crops. But why it should be called GMOs? If you have the base editor that I give you example, and CRISPR-Cas9 construct present in the genome, not removable, then it is called GMOs. If you have transgene present, but if the transgene is removed already, and you have only mutation present in the genome, but no foreign DNA inserted, then it is not called GMOs.
Sir, one more question, sir.
Yeah.
So when we have very well established RNA technology to develop a new traits in the organism, now this we are having CRISPR-Cas9 technology, newly developed one. So which one is the most better one?
Just repeat the question, please.
We have established the RNA technology to incorporate a new trait into the organisms, right? Now we have CRISPR-Cas9 technology. We are also doing almost the same thing. So which one is the better one, and which one give the better result?
You know, I understand. So RNA technology, I mean, you are talking about transgenics and all these things, huh? Yeah.
Yes.
So for transgenics, you need to incorporate a foreign gene into your cultivar or into your variety. Exactly. And that gene need to be present constantly in order to show the desired result.
Exactly.
So you cannot remove. You have to cultivate the plant with the foreign gene inserted. That's called transgenics. Okay.
Yes.
Now, for CRISPR-Cas9, for few means most of the application where you can incorporate the CRISPR-Cas9 in first 1 or 2 generation, and once the target mutation is done, you can remove the CRISPR-Cas9 construct, so the foreign gene need not to be present there. Okay.
Oh.
So this is the basic difference. And now another thing is that you can modify the endogenous or native genome of your plant by using CRISPR-Cas9. But with transgenics, genome level modification is very difficult. Okay.
Yes. Thank you. Thank you so much, sir. Thank you so much. That's really very, very good information.
Dr. Umesh?
Yes, yes, ma'am. Am I audible to you?
Yes, we are.
Yeah, thank you very much. Actually, maybe I was having problem with my system. And really, first of all, because I'm also in university and I'm also a plant scientist. I was working on wheat initially in my PhD and then moved to potato and other plants, rare plants. Okay, actually, I was listening that presentation and I have one query in terms of, because other participant also raised that question about that single gene, and if it is already characterized, we are having that information. So that is sure that if we are talking about the quantitative traits, that's not the tool to play with. If I'm talking about that in wheat bread making quality, that is a quantitative trait, so many QTLs that is going to control.
Yeah.
So we cannot touch that part. That's sure that with quantitative traits that CRISPR is not the better tool to play.
Yes, yes, you're right. Absolutely
Okay.
right. For quantitative trait, I think manipulating a quantitative trait with CRISPR-Cas9 is really, really difficult. If you have a monogenic trait, a single gene is controlling a trait that is qualitative, that is easy to manipulate with CRISPR-Cas9. I totally agree with you. But how the pace of development of CRISPR-Cas9, if you have read this paper, from Jack Lipman's group from Cold Spring Harbor Laboratory, they have edited, promoter of some homologous homeobox genes of tomato.
Okay.
And quantitative trait is being manipulated with CRISPR-Cas9 by multiplex editing. Okay, by rearranging the cis elements of different development related genes. So I can forward that paper to you. But
yeah.
I totally agree with you that it's very difficult to go for quantitative trait with CRISPR-Cas9.
And then my second question in continuation of that, if we are talking about that, we are going to compare that initially people were working on transgenics and now they shifted to that CRISPR. If I'm going to compare whatever that in one box, that existing technologies, whatever conventional, move to the transgenic / CRISPR.
Yeah.
In terms of cost, is there any cost significant?
Oh, yes.
Because
yes,
always
lot of reduction. CRISPR-Cas9 can be done anywhere in an undergraduate college. Okay, only one tissue culture laboratory is needed.
Okay.
And for transgenic, you need to validate your plant with southern blotting, western blots, northern blot. Okay, lot of expensive experiment. But here only you have to do only transformation, grow the plant, and then amplify the PCR, send for Sanger sequencing. You get to know whether it is edited or not. That Sanger sequencing per sample, it takes 300 rupees.
Yeah. Okay. Now, one more question. While we are talking about that, we have edited the gene, and now we are moving to the field, and we are going to grow a crop. For how many generations that this particular editing will be fixed? Because with transgenics, we are going from T.
Yeah.
Whatever that stability of that editing.
Yes, you're very, very right. So, once it is happened in T0 itself, means if you could detect your mutation in T0. So for my case, in the base editing example that I showed you, in the second generation itself, we got homozygous edited, means both the allele edited. Okay. So, means for the students, a gene has 2 copies, and this 2 copies in case of diploid organism, and each copy is called an allele. So both the allele is edited in the next generation in T1. Maybe in T0, one allele is edited, but in the next generation, we got both the allele edited.
Okay.
So it is very high fixing rate. Yeah.
Okay. And now the question from student and we faculty both, because this is the even third training, that theoretical training, I can say that I have attended one Gyan workshop on that CRISPR also. How we people can train ourselves for this gene editing or CRISPR, because while we are talking about the training with that ICGB and other institute give a very high cost, 20,000 and something for 10 days. So what better we can get placed to get the better training, because we have to start theoretically, we are going to get updated ourselves from literature or whatever we are attending these lectures. But on working bench, we have to train ourselves that what we can do for that, because every student or whatever we want to start that part. So how we can do it?
This
We are ready to pay also. We are ready to pay also. I'm not saying that free of cost.
No, not free of. I think DST fund for Karyashala. It is called Accelerated Big Gun, and they fund up to 5 lakh rupees for doing high-end workshop. But that is sufficient. We can even freely train some participants, but the participant limitation, of only 25.
Yeah.
So 25. Suppose all of you, if you apply,
maybe maybe Dr. Vish can get in touch with Dr. Kutubuddin to find out the best possible and the low cost way to train.
Definitely, ma'am. Definitely. I'm quite desperate to learn these things.
Yeah. I just have one last question of the session. Thank you, Dr.
Thank you very much, ma'am. Thank you very much, sir.
That it is just my curiosity that when we are comparing the RNAi, like it is a post-transcriptional gene silencing. So basically, the idea is we are targeting whatever gene, but it should be the exons. The double-strand RNAs should be complementary to the exons. Means they should be covering the exonic portions, not the introns, not the promoters, not otherwise.
Yes.
But in case of CRISPR, though we are also targeting some portions of the genome, but considering that we have to keep in mind the PAM sequence, which in your opinion should be can be safely covered, whether for eukaryotic system, the intron, exon, or any specific and closest to the PAM, means all those things have to be taken care. So which in your opinion would be best to work to
you're asking my opinion on exon or intron?
Yeah, exon. Which
obviously
exons.
And exon, even not the exon that are present at the end of the gene.
In the beginning.
First 1, 2 is better. And what we do, I can share it with you. What do we do? We just take the protein sequence first, and we do the domain analysis on which domain is important for the function of that of that gene, and then we determine that if we come back to the sequence, we determined which portion of the exon is contributing to that domain, and we especially target that region.
I think the best approach would be the earlier exons, which you have already.
You're right.
That would be
earlier one base pair or even one base pair deletion would cause a whole disruption of the.
But suppose there are triplet. If it is a triplet, then might not be. So we need to take care of that.
If it is triplet, it is not. It is not
going to hamper. It's not going to hamper the frame. Yes. So we have to think on that also, that we should be careful on those points.
Yes.
I think we need to now. Yeah. Thank you. I think we need to now close the session because we are already running. We have run into the second session, but it was really wonderful talking to you, Dr. Kutubuddin, and it has been a great pleasure that you have given your valuable time and also patiently addressed all the questions. I think the question and answer was pretty long because I find that the plant lectures are driving a lot of curiosity, and it is so applied, and so much we can relate to our daily life, that plant sessions have been very, very interactive, and people, all participants could relate. And on this, I once again congratulate you for all your, and I hope that you have even more inventions to share with us. So many, you have shared so many unpublished results, and it was really fascinating, and it was fantastic listening to you. And you just kept your word that the stories that you had to narrate, they were really fascinating. Thank you so much. And I really wish you all the good luck in your future endeavor. And on behalf of our university, Maulana Abul Kalam Azad, University of Technology, West Bengal, I once again express my gratitude. Thank you so much for a wonderful session.
Thank you very much for inviting me and giving a chance also, because it was my pleasure. And I would love to share those articles if you want to share with the students, because they might not have access. And I would be also
Absolutely.
I would be also love to be connected with Dr. Manoj Kumar for any future collaboration.
Yes.
He is in that field for a long time.
Yes.
And yes. Okay. Thank you very much.
Thank you. Thank you so much. Thank you. Have a nice day. Thank you. Yes. So now we will move on to the final session of this faculty development program. And I can already see our next speaker, Dr. Ranjita Biswas, already there online. And I welcome you, ma'am, to this session. So to all the participants, I hope you are all there. You have not left. We don't have any break now in between. So we will break immediately after this session. So keep your patience. And let us now start with the last session of this faculty development program. So in this session, we have Dr. Ranjita Biswas, our very close friend and colleague of ours. She is the associate professor in our Department of Biotechnology, who is actually working on CRISPR technology. So I found that we can have someone from in-house to really deliberate on this session. So welcome, Dr. Ranjita Biswas, and let us now, I will have just briefly introduce you to the audience. And as you know, this is a faculty development program focused on the CRISPR-Cas technology. So you will definitely have a very good session with them, with your research. Now, Dr. Biswas is she is going to talk on something that was less touched upon. Yes. So today, as far as her research career is concerned, she had been earlier, after she completed her PhD from IIT Delhi, she moved on to Bioenergy Science Center in USA, Oak Ridge National Laboratory, for her postdoctoral training. And actually, she was working on some anaerobic thermophiles, bacteria. So these bacteria, they are efficient in producing ethanol. Okay. From other biomass, lignocellulosic biomass. So this is something very interesting. So the bacteria is being used for the production of some valuable, some commercially important product. And now she has, following that, she was awarded the Ramalingua Swami Fellowship, and then she moved to Macau in our department, and now her lab is actually focused in engineering the metabolic pathway of certain thermophilic bacteria, and for biofuel production. You have already heard Dr. Amit Ghosh talking about a similar type of topic he had, and where you saw he used the yeast as his model. But today you will hear from Dr. Bishash on thermophilic bacteria. So her topic today is utilization of CRISPR-Cas genome editing for thermophilic microbes. So you will have a very good session, I believe, Dr. Bishash, with the participants. And obviously, since this is a kind of a technology which we are all now glued into, trying to find out how much we can improve our lifestyle, our living style, our life. So I think with your talk, we can have another sense of some positivity. So with this few words, I welcome you on behalf of our university. You are our own member. So once again, thanks a lot for accepting the invitation. And now the stage is yours, Dr. Bishash. Thank you.
Very good morning to all the audience, the participants, and thank you, madam, for the introduction. And so I just got to attend a bit of the last session. So the participants are very enthusiastic, and that's a very good sign. So we are all going to have a good and a long discussion on the various topics. So yeah, this is a very interesting toolbox, that is the CRISPR-Cas. And a lot of work has been going on in the past, maybe like it's less than a decade that we have this toolbox with us. So today I will be talking about this another aspect. You have been, like, all the sessions has been organized very well, and we had covered variety of topics where CRISPR-Cas has been introduced. So today this particular microbe, that is the particular category, that is the thermophilic microbes. So let me just start from where this research actually happens. So my lab works on various kinds of developing various kinds of technology for industrial applications. So the challenges are twofold for us when we are developing certain technology, where the implications are commercial. So first part that we have to delve into is the suitable biomass, and then finding an efficient microorganism. So suitable biomass is also a big challenge, that we should not be competing with the food crops. So whatever biomass are available with us, especially the agricultural residue or something maybe from the feedstock, or maybe the waste that has come from the forest resources. So food crop needs to be avoided because there is a limitation of land and water. So we don't want to compete with food crops. So although there is a lot of success story on ethanol production from corn, and from sugarcane, you have the corn is coming from the US model, and the sugarcane as a feedstock is coming from the Brazilian model. So it is a very successful commercial venture, but now everything is suffering from a sustainability issue. So because of that, now we are back into the lignocellulosic feedstock. So the main challenge is a cost-effective deconstruction of the recalcitrant plant cell walls to liberate the fermentable sugars. So this recalcitrant nature of the cellulose, the hemicellulose, the lignin that is making up the lignocellulosic biomass is the main challenge. So how to get the sugar out of it? So some groups, the leading groups in the world, some are working on this part, that how to make the biomass suitable. So like you heard our previous lecture, that can we have some transgenics? So in this regard, the transgenics which are having less lignin content, so that the enzymes are more accessible to the cell wall, and we can have an easy liberation of sugar. So that is one strategy. Some groups are working on that. And the next part is the efficient microorganism. So can we find a microorganism which will be able to utilize the lignocellulosic biomass directly? So yes, in nature, such microorganisms are available, but they suffer from low yield, titer, and productivity. So the terminology yield, titer, and productivity may not be very familiar to all, because that is how we talk about when we are defining a bioprocess in terms of commercially viable or not. So yield, just briefly, it is all about the conversion, that how much sugar or how much biomass you feed to a microorganism, and how much of it is getting converted into the desired product. So normally we show it by gram per gram. So gram of product per gram of substrate. So that gives you the yield. So titer is also similar, like suppose I'm producing ethanol, that will be gram per liter or milligram per liter. So it's kind of a, you can say it's concentration. And productivity will be similar, but it will be having a time factor now. That how much of a particular, let's suppose ethanol you are producing in how much given time, whether it is taking 1 day, 24 hours, whether it is taking 7 days. So a process is defined by all this. So a commercially viable process needs to meet all these requirements of yield, titer, and productivity. So these are the challenge. Although we may design a microorganism which may be very good at a laboratory scale, it may give a very good result in the test tube, but it may not work in that manner at a fermentation level, at a bioreactor level. So it will not see the daylight to become a commercial strain or an industrial strain. So keeping all these aspects in mind, that you have to have a meat, you have to reach a place where you find a suitable biomass and you design an efficient microorganism, then only we can propose some industrial process or some viable technology that will have takers in the market. So keeping this background in mind, so this is also a same depiction of what I have been telling, that you have a lignocellulosic biomass, it needs to convert it into sugar, and then we have to find a suitable microbe to convert it into a fuel. Now, just a glimpse of the kind of work that my laboratory undertakes is a biorefinery work. So if you see this hypothetical tree, this is actually based on a report, that is the billion-ton report. The billion-ton report was published by Department of Energy from US in 2011, and some update also came later that simply emphasized that every year we are getting around 2.5 billion tons of raw material, or you can say the waste lignocellulosic material is available on Earth, and that can be utilized for a bio-based industry. So green technology is very much a possibility in a sustainable manner with so much biomass available. Now, when so much biomass is available, it becomes an environmental issue. Then is everything going to compost? Now, composting is also a challenge. It takes a lot of time, although it will add the green material back to the earth, but it is not a overall solution for the challenges that we face. So if you just look at this tree, so that is the whole concept of biorefinery, that you have the various kinds of biomass coming from various sources, even the rice straw, wheat straw. So that is a major problem in our country right now, the major pollution threat because of burning of all kind of stubbles all around from the agriculture residue. So we heard all the stories about Delhi, northern India facing all kinds of problem. Not only that, even Bengal, even Calcutta is facing, Kolkata is facing so much of pollution problem. So we have all this. So even if you see the farmlands in Bengal, there is ample of biomass burning. So those are all our bioresources. So if you are able to break this part, then it can go into enzyme production, ethanol, butanol, fermentation. We have various kind of hydrocarbons, synthetic biology. Everything is now based on this kind of a bioresource. So with this background, again, a little bit of this, so that if you have so we can have a two-prong approach, that we are modifying the tree as well as we are modifying the microorganisms. So this is a further depiction of the same concept that my laboratory is trying to emphasize. So in this direction, there is the availability of certain microorganisms in nature. So let me emphasize one thing, that is why a thermophilic system. So you have heard Dr. Ghosh, Amit Ghosh, 2 days back, where he's working on a yeast strain, and yeast is a industrial strain. A lot of work has been going on, and it has successfully produced many chemical compounds of bio-based origin. The only limitation with yeast is it is a microbe which can only utilize glucose. But when we break down biomass from plant source, we are not only getting glucose, that is your hexose sugar, but we are also getting pentose sugars. We are getting xylose and a lot of other kinds of sugars. Then what to do with those? Then it becomes such kind of system which is very expensive. So when you see ethanol production, for example, biofuel production involving yeast, it is expensive just because there is a saccharification step where all the biomass needs to be converted only into sugar, that is your glucose. So rest of the sugar needs to be removed. And that saccharification step is expensive because of the involvement of expensive enzymes. Enzymes are very expensive. So that's why, although India has a mandate that we will be mixing ethanol in the petrol around, it should be like 20%, but we are far behind. It is less than 5% right now, okay? Because we do not produce so much of ethanol. And so much of ethanol we can only produce when we have such kind of microorganism which is able to utilize all the plant sugars. So hexose, pentose, arabinose, xylose, whatever is listed, it should be able to utilize. So one such microorganism is Clostridium thermophilum. So when we talk about thermophiles and mesophiles, so yeast is a mesophile and Clostridium is a thermophile. So thermophile has an industrial relevance. So when we are working at a large industrial scale, the initial biomass that we take, suppose we are taking wheat straw. So the wheat straw will undergo certain pre-treatment. So for pre-treatment method, heat is involved. So whenever you are heating a particular biomass for pre-treatment, then you have to cool it down to bring the energy, maybe the temperature to around 30 degrees if you want to use yeast. So there is a lot of wastage of energy and money. So the process are very expensive. So how to cut down cost? So we want to look for microorganism which can grow at high temperature. Why to cool it down? It takes a lot of fuel. Cooling and heating, both is a problem. So when you have a pre-treated biomass, so a strain like Clostridium thermophilum, which grows at around 65 degrees centigrade, this will be able to work faster. As well as at high temperature, the chances of contamination is less. So there are certain industrial implications. So we have, but there is a challenge with the thermophiles. Thermophiles are very robust because they are evolved in nature in such a manner that they are extremophiles. So their protein system is not like the ones for a mesophile. So they are very stringent. They have a stringent system of maintaining their genetic material. So whenever we try to manipulate it in some way, there will be a certain kind of a backup mechanism to revert it. So it is very difficult to work with such kind of thermophiles where the system is very enclosed. Okay, so when we started working with this particular, so this work I had done when I was designing this Clostridium thermophilum in my previous laboratory. So it is an anaerobic system and they work directly on the lignocellulosic biomass. So it will sit directly on the surface of a rice straw, wheat straw and start degrading it. So the saccharification step, the addition of enzyme step is removed. So it makes it a very interesting microorganism and it is producing ethanol also. Now the problem is it is making low concentration of ethanol and also the tolerance is very poor. So keeping all these challenges in mind, now we are set to design the pathway. So what will be the ideal designing for this? So it is making ethanol, producing ethanol, lactic acid, formate, acetic acid, some amino acids, as well as it is making some hydrogen. So when glucose molecule, here the glucose is shown just to elaborate on the pathway. Otherwise, it will be utilizing the xylose also. So it is utilizing directly the lignocellulosic biomass involving glucose, pentose, everything it can digest. So just for the convenience sake, this glycolytic pathway is shown here, that how it will be moving through, the carbon flux will be moving through this pathway towards ethanol fermentation. So here idle or micro for industrial production will be that if you see this red crosses, that means I want to delete the hydrogen production. I don't want any electron getting lost as hydrogen. I don't want any carbon getting lost as lactic acid. So this needs to be deleted. Formic acid pathway needs to be deleted. Acetic acid pathway needs to be deleted. So I am looking for 5 deletions. So here I have shown 2, although it is hydrogen, but I will show a little bit of this, that there is a 2 type of hydrogenases. So in this 2 type of hydrogenases, there will be 2 requirements. Overall, there are 5 hydrogenases, but 2 deletion will be enough to knock them out. So if I'm introducing 5 deletions or 5 pathway deletions in this particular host, I should be able to direct, if you see this particular flow, I should be able to direct all the carbon flowing towards ethanol. So this is the ideal situation for me, that I have to metabolically engineer the pathway in such a manner that everything is going towards ethanol formation. Now here starts the challenge, that how I am going to design this microorganism. So what are the options available to me? So when I started this work, so that time CRISPR-Cas was just making a beginning. So it was not a very sought after or talked about toolbox. So it was the very beginning of this work. So that time the homologous recombination appeared to be a best strategy. Although, so if you see here, I've shown that these are the hydrogenases that are available. So if I am designing the plasmid in such a manner that I am able to remove 3 hydrogenases. So overall it is 4, not 5 like I mentioned, it is 4 hydrogenases. So these 3 will be deleted in one go by one deletion. And the ECH hydrogenase, that is the nickel NIFE hydrogenase, that will need a separate deletion because of its location. So the best part in this was that we were able to delete this hydrogenase by just inactivating the hide G. So it has a combination of hide E, F, G. So this, all the subunits needs to be put together to have an active hydrogenase. So by just deleting hide G part, this particular subunit was common in all. So just by deleting one subunit, we were able to knock off all the other hydrogenases. And for this particular one separate was designed. So this is already a published work. You can see the reference and you can also read the paper for all the details. I'm just giving a glimpse. So we will start with the CRISPR. So if you see this, when this designing was done, when this knockouts were developed, it was a very laborious, tedious, it took long months to work on such kind of plasmid systems. And we were using merodiploids because here for when you're working with thermophilic systems, it is very difficult to get a plasmid integrated. So although we designed a plasmid and that particular plasmid was used, but to have a chromosomal knockouts. And so we had to rely on availability or this particular design in which, if you see this, merodiploids were designed. So I have not given a detail of this here because it is a very long work. So just if you are interested, I can give you the reference if somebody is wanting to work in such a manner. So here we were having 3 sets of primers and 3 kinds of amplifications were checked to verify that yes, the deletions are incorporated, and we were able to make successful mutants out of it. So this was a homologous recombination strategy. By this, the lactate, then your hydrogenase, then the formic acid, acetic acid, all this were deleted. So final result were around, we got 70% improvement in the ethanol synthesis. So 70% is pretty good using such a microorganism in which 5 metabolic pathways were shut down. So when I know most of you are working in this field. So, whenever we are shutting down a particular pathway, it becomes very difficult again to make that microbe work in a similar manner. But we did certain kind of fermentation strategies also we used. By that, we were able to obtain 70% of the theoretical yield. So this was a long work which involved 3 years of work. And finally, this was the outcome of this work. Now, like I have emphasized, that this homologous recombination toolboxes are laborious. So the kind of work that I do where we have engineering the pathways. So we want to shut down entire pathways, and sometimes there can be 4 or 5 pathways which are interfering. So having homologous tools are always not the answer because of the time involved and the deadlines of the project. So, and in the meantime, this CRISPR-Cas strategy is also becoming very viable. So the group, so before I introduce this, so you have been listening to the lecture for last 5 days now, so you are very much aware. So this is just one glimpse from a Nature publication that it's a beautiful design here. So you can see how it can be utilized, the CRISPR technology for gene editing, gene regulation, epigenomic, chromatin imaging, then your base editing, RNA targeting, chromatin loop topology, chromatin imaging. So the limitation, the possibilities are vast, and various groups are working on various kind of implications and applications of this. So my group, we are working on the gene editing part by using this tool. So again, I'm touching upon this because in the last 5 days, I think you must have already seen a picture like this where we are talking about what is the mechanism. So I'm not going to spend much time on mechanism. You must all be knowing by now. So there is a part of adaptation, there will be a part of expression, and there is a part of inference. So by these 3 parts, we are going to use this or design this toolbox and feed the kind of protospacer that we need. So this protospacer, how efficiently can we select the protospacer and whether, so suppose you are having a system where you are trying to do an endogenous system. So a heterologous system can be there or an endogenous system can be there. So if you are having an endogenous system, so the designing can be a bit tricky just because that you are now having, you have to be careful that what kind of protospacer you are choosing, that whether you will be designing that it is targeting itself, or sometimes there is a problem. Like in our particular case, we are working with another strain that is Thermoanaerobacterium. So right now the lab is working, have a project on this. We have a project on Thermoanaerobacterium. So in this particular strain, for one set of gene, there are copies, there are several copies. So I have to know that what kind of protospacer we are designing and when this particular copy will be available and when this particular, and they are not available on the same chromosome. So they are scattered. So what will be the strategy and which particular one do I want to knock off? Or am I wanting to knock off all, or knocking off all is not required? I want to have a weak expression. There may be some expression, but I'm not trying to shut it down completely. So these are the layers of complexities that are introduced once we start seeing multiple copies of a thing. Now, so just a brief touch upon on this, that the Cas proteins obtain and store segments of the invading viral genome called protospacers. By directionally integrating them into 2 adjacent CRISPR repeats, the protospacer will serve as a memory henceforth. That is your adaptation phase. This is what the nature is doing. And now we are using this kind of a tool that is naturally available to put our protospacer of interest. Now, a CRISPR RNA complementary to the protospacer is synthesized from the CRISPR loci, that is the expression in the maturation phase, which then combines with the Cas proteins and recognizes its complementary sequence with or without the protospacer adjacent motifs. Now, PAMs are important, and additions to adjacent to the CRISPR. So when we are working with Cas9, this is important. But when I'm working, but in thermophiles, as we go along, I will tell you, Cas9 will not work. So that is one limitation. Although a lot of work has been done on Cas9, but Cas9 is not available for thermophiles. We cannot use it. Okay, so I will let you know why. And this is then followed by the CRISPR RNA directed cleavage of invading RNA DNA by Cas proteins. So this is our interference phase. Okay, so we'll be going to the last, that is the cleavage part. Okay, now let's have a brief introduction also about this, that how many types of CRISPR-Cas systems are available. So we have class 1 and class 2. So under class 1, there is a type 2, type 1, type 3, type 4, and under class 2, we have type 2, type 5, and type 6. So normally for the Cas9, we are talking about the type 2. So, and the classification is by the virtue of their 3 unique signature genes, that is the Cas3 in type 1, Cas9 in type 2. So you all heard about Cas9. So all the lectures, most of the lectures were talking about Cas9, and Cas9 is a well-defined toolbox. It is applicable in the mesophiles. So this is another arrangement. If you see the phylogenetic tree about how their origin is, if you want to know about the evolutionary pattern of this, so just you can have a look at this. So now let me move to the main part of our work, or main part of the interest area that my lab is working. So Cas9 is not active above 42 degrees centigrade and cannot be used for genome editing in obligate thermophiles like C. thermosilum. So Clostridium thermosilum is one strain which I worked for ethanol production. Now, while working on that, so another strain was discovered. So that is a Thermoanaerobacterium virendrai. So my lab currently is working on that strain. So this strain is also growing at 55 to 60 degrees centigrade. So now this Cas tool, so CRISPR-Cas9 cannot be utilized. So let us see what are the options available. So this is one strategy. So the strategy to design CRISPR-Cas system for Clostridium thermosilum. So in this, so since Cas9 is not available, so certain other test or strategy has been used. So it is work in progress. So I'm not saying at the end of the talk that it is successful, but then it is not completely proven. Okay, it is work in progress. So, but it is showing a potential results that yes, it will work. So there will be a selection of native type 1b and heterologous type 2 CRISPR system for thermosilum, Clostridium thermosilum. So this work has been done not in my lab directly. So this is my collaborators from Thayer School of Engineering near Boston. So this is my group where I was working for my postdoc too. So I worked on the homologous recombination engineering for Clostridium thermosilum. Now the work has been forwarded with the CRISPR-Cas system. So this is their work. So I'm just sharing you what kind of strategies followed. And towards the end of the talk, I'll be talking about what my lab is working, how we are doing for the thermoanaerobacterial system. So this has now said that in this Clostridium thermosilum, we have options. So we have the native type 1. So there are 5 CRISPR available in this Clostridium thermosilum. There are 5 copies of the type 1 available. So out of that, only one locus is having the complete CRISPR system, CRISPR operon, and the rest 4 are incomplete. So they cannot be used. So that is the type 1b is available. And then one other is chosen, that is type 2. So type 2 is your Cas9. Cas9 system is also chosen, it will be heterologous. So the strategy they have used is, it is a one-step, they have tried to design a one-step CRISPR-Cas genome editing system, and another is a two steps that, so it is showing much better result as per their report, that it is if you are doing a two-step CRISPR-Cas, two-step is we are having a two plasmid sets they are taking. So first, the one plasmid set is introduced, and then the second plasmid set is introduced as a counter selection. So that is the simple strategy. But when one-step CRISPR-Cas genome editing system was engaged, there was certain kind of knockouts were visible, but the CRISPR operands were leaky. So in the expression, so there were not complete knockouts. There were some leaky. So during the transformation in the plates, we could see some colonies, although those colonies should have not been there, but it was not a complete system. So the answer was not there. So in that case, one recombinating system was developed. So this recombinating system, to overcome the limitation of the homology directed repair. So a small recombinating system, so I will tell you about that, was introduced. And by that, one was able to step into the two-step CRISPR-Cas system. So two-step CRISPR-Cas system was able to show a clean knockout, but it was not 100%. So because of that, this strategy is still in work in progress. Okay, so we will just take a look what was the main issue in this. So if you look into this, so this is your lignocellulosic biomass, so CRISPR-Cas editing in Clostridium thermophilum. So this is the strategy that from your grass or direct wood, so there is no conversion of sugar step, it goes to Clostridium thermophilum. And now this goes into a consolidated bioprocessing. So people who are not from this background of fermentation, so this consolidated bioprocessing terminology is very simple, that you take one container, like this is one bioreactor, you feed the grass or feed the wood chips, whatever, and put the microorganism. It should be able to digest and hydrolyze and do the bioconversion and produce the fuel or chemical directly inside one vessel. So that is known as consolidated bioprocessing. That's it. So in the native, so in the native strategy, so we have the type 1b and the heterologous type 2. So in the native type 1b strategy, using the plasmid toxicity assay and the palm depletion library results. So just go through the paper also. The material method is too exhaustive. So in this time limit, it is not possible to show everything. So I'm just telling you the main glimpse of this work. So the RNA designed the guide RNAs by identifying a strong PAM site on the sense strand, okay, in the PYRF locus. So this particular locus, you can check, the details are given there, that this particular strand, that is 1266, this is the primary native segment that we are taking without any kind of manipulation in there. And then use the 37 base pair sequence immediately downstream as a target spacer. As a control, a 37 base pair on the sense strand in the PFI locus without a predicted PAM site, and then use this for the non-target spacer. So we have a target and a non-target spacers. So in general, 37 base pair spacers were immediately downstream for strong predicted PAM sites were used for the target single guide RNA, and non-target single guide RNA were the same length but lack predicted PAM sites. So there are so when the PAM depletion library was created for this particular microbe, in that there were certain predicted PAM sites and there were certain non-predicted PAM sites. Okay, so around 10 were identified, which were having a very high expressions. So with those high expressions, this experiment was conducted, but you can have a design in which you have a list of the PAM sites. So we will see later that this
Hello?
PAM sites. Yeah. Hello?
Hello.
Yes.
Yes, ma'am. Ma'am, the slides are getting blurred. Is it there, ma'am?
Ma'am, please unmute, ma'am.
Hello?
Yes.
Yeah,
Yes, ma'am.
Ranjita, can you
Is
hear
there a
me?
problem? Yeah.
Yes.
Yes.
The slides for me, I cannot, I don't see any movement.
Ma'am, it is getting blurred, ma'am.
Blurred, it is getting blurred. Can you just,
ma'am, you have to reshare it,
yeah,
ma'am.
reshare it. Yeah, just reshare it or stop.
Without optimizing the, ma'am,
Optimizing
optimizing screen.
Yes, the network issues could be there.
Yeah. But it is showing full strength, the network is showing. Okay, so you want me to stop and again share?
Yes, that would be better. Yes. Please. I also
ma'am,
got.
if there is internet issue, you may switch off video, ma'am, at the time of deliberation, ma'am.
Yes, that would be better. Ranjita, you can turn off your video.
Yeah.
Even I think I also have to turn off my.
Right. I think it's creating problem.
You have to just. Not yet.
Is it okay now?
No, not.
Yes, ma'am. Now it is okay. Yes, ma'am.
Can you go back? 2-3 slides? Yeah, from here onwards, you can just. I think will it be okay to the participants if she moves to the next slide? Yeah, you can start. You can continue from the earlier slide.
Yeah. I will continue, and we can go back and forth.
Yes. And Prem, can you mute the others?
Dear participants, is it okay with the slides? We will be proceeding with this.
It is clear now.
Yes, ma'am.
Sure. So you can. This is clear. Rajita, you can continue.
Okay. Yeah. So one was the native type is already available. So this is a heterologous type 2. Since Cas9 is already characterized, well established, well proven in various systems, so Cas9 remains a favorite for all. So now the only challenge that remains how to have a system like this in a thermophile. So after going through the database, so we came up, which were like we had certain like the geobacillus and 2-3 more the Thermoanaerobacterium saccharolyticum. So we were able to find some in this particular similar kind of segment is available, if not similar, but it can be defined under the type 2. So taking those, this particular heterologous type was selected and designed. So in this, a single guide RNA was designed using batching software. Briefly, we can say that using the CRISPR, we designed the guide type 2 single guide and the reference genome of the Clostridium acetobutyricum. The guide length was set to 22, 24, 20, and the time site was set to this particular between this range. And from the the name is given, so based on the microorganism. So one is the Cas9, ACE-Cas9, and Thermo-Cas9 respectively. Only single guide RNAs with 40 to 60% GC content were selected for further testing. So lot of basic screening was done. So one was selected finally. Finally, the ACE-Cas9 was found to be effective. If you see the results, the upcoming results. So this strategy was a bit. So in one CRISPR-Cas genome editing, in the time being, the plasmid toxicity assay was modified to include 1 kilo base pair template targeting this particular locus, that is PYRF locus. So this PYRF locus, I can tell you one thing since we are hearing this. This is the gene encoding orthodentine 5 monophosphate carboxylase. So when we are using this, so we can do the selection on the plate using this is your this is an analogous of uracil. So this analog of uracil can be in the plate, and then if the deletion is there, then it will not be able to grow. If the deletion is not successful, it will be able to grow on that. So that, and this particular deletion is not having any role in the microbe itself. So it will not affect its other factors of microbe. So that's why typically when we work with the Clostridium thermophila, whether we are working with a homologous recombination method, the traditional method, or in this CRISPR-Cas method, we use the same marker, that is PYRF deletion. So one version of the plasmid expressed as single guide RNA targeting this particular locus at the site with the predicted PAM sequence, and a second version expressed a control that targeted this locus at a different site without a predicted PAM sequence. So the strategy remains the same in both. The one-step plasmid with a target or a non-target single guide RNA was transformed into this particular strain. This is our the selected strain with the promoter this driving the Cas operon expression and recovered in the CT food is the media normal. This is your rich media that we use, and it was grown for overnight at 50 degrees. So little bit about the methodology. And in the type 2, the plasmid used for the one step itself. So the plasmid used in the toxicity assay with the addition of this. So I think I will not focus much on the strategy on the material method part. You can refer to this paper. It is a publication in Metabolic Engineering 2020. So please refer to for all the detailed methodologies given there. So let us go to the result, what happened actually in this. So what was the, so first identifying the PAM sequence, then the PAM identification was to try to get the original sequence of the invading elements that ended up as a CRISPR spacer in the Clostridium by blast search. So this particular PAM sequence was taken, and in the presence of a strong promoter and with the PAM sequence of 50 50 CAG TT and this GAT CA30, we observed it was observed a 10-fold decrease in the transformation efficiency, indicating that so PAM assay with 10 showed ASSA10 showed strong deletion with both spacers, and can be summarized that the degenerate sequence of TTN30 and 50 TNA. So finally, this was the one that could be selected. So this the PAM selection. Now, after the PAM selection, one toxicity assay was very important to determine the CRISPR-Cas activity. So if you see this with the plasmid toxicity and the chromosomal toxicity assay. So if you just go to the table, in the type 1B, and you have the Cas promoter. So if the native, with the native, when the plasmid target is, we can get a result, but with the, when we are doing the plasmid toxicity assay, so you can see a positive result. So we can see the type 1B, that when we are using the, we can see the Cas activity in the plasmid target, as well as with the this particular promoter also we can see the activity. But when we are going for the chromosomal target, not the plasmid, so if you see the chromosomal target, in that case also we can see the activity here. And when we are taking this particular Cas promoter from the Thermoanaerobacterium saccharolyticum, so you just see the fourth marking. So in this particular, your guide RNA promoter and the Cas promoter is coming from T-Sac. So in this case, you can see the chromosomal target. It is working for that, but for the plasmid target, it is not able to work. And when you see the chromosomal target, again for the chromosomal target, so all are effective here. So we can see that for the chromosomal target, this particular promoters is working. So the first part is that in the plasmid toxicity assay, we were able to see that yes, it will be effective. So the initial signals were fine. So with this initial signal, we move to the next part of this. So the toxicity assay demonstrated a highly active engineered 1B CRISPR-Cas system and a highly active type 2 Cas9 system. So these two results were we could get from this all the various analysis that was done. So the key differences that came between the native type and the heterologous type, that one is your Clostridium thermophilum is the native type, and the Geobacillus stereothermophilus C9 Cas9 seems to be working here in this case. So this may not be when you actually match it with Cas9 of Mesophyte, it does not have the same kind of you can say the similarity is not 100%, but it is having similar kind of model. So finally, we are placing it here. The label will be Cas9. It is not the general CRISPR-Cas9. So the PAM sequence you can see for the native type, only this sequence are effective, and for the Geo9, this PAM sequence will be utilized. And the guide RNA, you can see this 97 nucleotide spacer. And for the type 2, 140 nucleotide spacer is okay. And the homologous enzyme is 4 protein. So we have Cas4, Cas5, Cas8, and Cas9. So this is the one that will be used for the single guide RNA. So, and here for the Cas9, it is okay. Only one protein is there. So in nature, native type, when we are using, so if you want to use the native type, then we will have to see that we have this 4 complexes coming together. Now, so making the type 1 system functional required identifying the correct PAM sequence and placing a strong constitutive promoter upstream of both the chromosomal Cas and the plasmid encoded single guide RNA, and making the type 2 system functional required identification of a thermostable Cas9 protein and putting a strong promoter in front of both the Cas9G and single guide RNA. So that is evident from your table, and this is what. Now, once the CRISPR-Cas genome editing system is effective but not efficient. So like I told you in the first part, that although it is working, but there are certain leaky mutants. So because of those leaky mutants, so if you see the transformation efficiency with the non-target and the target. So non-target is fine, and with the target, you can see with the type 1B, we can still get some transformation, some colonies we can get in the type 2 with the target. Yes, it is showing 0 here, and in the type 2, one Nikis is also used. So that detail we have not explained here, so that you can see in the paper itself. So here we will just focus on the main part of this. And the percentage increase observed in this plate, when we are seeing the plate, in the non-target, so if you are checking in terms of the phenotype, so in non-target, you can see there is still some colonies are there. In the target also we can see. So overall CRISPR-4 change that is happening is the 1.3 and 7.3. So it is working, but it is not really giving the kind of result we are expecting for 100%. So the limitation of the homology directed repair in Clostridium thermophilum. So homology directed repair and how the attachment is going to happen and how it is going to identify the same base pairs, like most of you are aware of the homology recombination. So we are not going into that. We'll just overall see what happens. So how to overcome the limitation directed repair. So combination of the high cutting efficiency from the killing assay and the low genome editing efficiency from the one-step editing protocol indicated that homology directed repair was the rate limiting step. So although we are seeing a high recombination, that is your combination of the cutting efficiency, but the genome editing efficiency is low. So once when it is done in the plasmid, it is fine. But when it is done in the genome, then the efficiency is not that high. So that's why this homology directed repair. So when it is integrated at the homology sites, then also that is becoming the rate limiting step. So to improve the rate of homologous recombination, 3 recombinating machinery pairs were tested in Clostridium thermosilum, that is your beta exo isolated from Acidotheobacillus paldus, and then one was taken from Clostridium stercoreum, and another is taken from Geobacillus species. So this was again tested to see if we can have some kind of a recombinating machinery to boost it up. So the boost up, so there was an increase, there was a 35-fold increase in this homology directed repair. So if you, that is available for the ATC only. So we will be able to see
Sir, we need to actually
when we can have this. So we will be able to see the change when we are bringing this another added step into strategy. So efficient. So the final test that was done was efficient genome editing using a 2-step CRISPR-Cas and recombinating system. CRISPR-Cas recombinating. So we will have the first step involves transforming cell with the repair plasmid containing the repair template and recombinases. So if there is a requirement of a recombinases, a certain fraction of the population will incorporate the repair template into the genome. And the second step involves transforming the cells with the second plasmid containing the CRISPR machinery, that is your single guide RNA Cas9 for the type 2 system, if it is a type 2 system. Then the second killing plasmid is used as a counter selection to kill cells that are not edited by the first plasmid. So you will integrate the first plasmid, and then the second plasmid will be entering to, which will contain the CRISPR machinery. So this will used as a counter selection to kill the cells that don't have the first plasmid integrated. So it will be a very good strategy that you can first send them that whatever system you want, that those are integrated, but where it is not getting integrated, the homology model is failing, there it will be so those will be killed. So only the one that is successful integration, they will survive, and we will be able to introduce the protospacers. So that is a simple strategy that has been designed for this, but still it is work in progress, because in some situations it is not working, and in some situation it is showing good results. So the final that came out from this was improved genome efficiency, editing efficiency from 14% to 70% by the type 1B system, and from 0.21% to 94% for the type 2 system. So by this strategy, Cas9 from Geobacillus seems to be a possibility, and both systems enable strains to be constructed in about half the time. So one thing that happens is we will be able to reduce the time that is typically required for doing a knockout in a Clostridium thermosilum. So doing one knockout, if everything is going fine, all the system is perfectly developed, the marrow diploids are working, then it is typically 1 month it takes under very good lab conditions. Otherwise, with this system, we will be able to reduce the time to half. So yes, you can see it is not 100%. So that's why it is still work in progress. So that is the work that was of interest, and I wanted to share, because there is very, apparently it is very good work, because apparent I'm saying because still we are not at 100%. So yes, it has a potential that one day we will have a good system for controlling or doing knockouts in thermophiles too. So in this regard, my lab is currently working on Thermoanaerobacterium virendrai. This particular strain is a thermophile anaerobe, and it ferments both hexose and pentose sugars into butyrate, lactate, acetate, hydrogen, and N-butanol. So at Macaut, my lab is working on butanol production. So butanol is an advanced biofuel, and the microorganism, so this is a new isolate. And just to have a little bit more look about the commercial potential of this microbe, carbon recovery of this particular strain is 90%, where 55% carbon is getting converted to butyrate. So butyrate is a nutraceutical molecule. So that is very optimistic or very advantageous point about this particular strain, that it is producing a butyrate molecule with 55% carbon going towards that, and 21% to lactate, some to acetate, and some to butanol. So with this strain, although butanol production is our target, that is the advanced biofuel, but the toxicity of butanol is very high for microbes. So normally 2% butanol in the media will kill the microbes. So my group is working in all the strategies that how we can improve the tolerance of the microbes to at least 5%. So it won't be commercially viable if I'm not able to make the strain tolerate 5%. So any solventogenic strain needs to tolerate at least 5% to become commercially viable. So that work is also going on, and it is having a genetically amenable system. So because of that, we are now working towards the CRISPR-Cas. So just let me show you briefly the strategy that is being followed, like I showed already for the Clostridium thermophilum. So for Thermoanaerobacterium 2, it is making lactic acid. This needs to be deleted. And so these are our target pathways. The acetic acid needs to be blocked, and all the carbon needs to flow towards butyric acid. And from butyric acid, it will, there is a partial conversion here at butyryl-CoA. There is a split, some will go towards butyric acid and some will go towards butanol. During the fermentation, the butyric acid synthesis or the butyrate synthesis will take place first. So acidogenesis phase is there, and after that, there will be solventogenesis phase. So when the pH changes too much, then the system will move into the solventogenesis phase, and gradually it will start utilizing the butyrate also, and everything goes towards the production of butanol. So our basic target remains shutting down the lactic acid and the acetic acid pathway. So for this, we have already started to work, and the whole genome has been sequenced for this particular microorganism, since this is a very new discovery. So we have got everything done for this in terms of all the genetic and genome information we have. So this work is taking us towards a CRISPR-Cas strategy. So we have a type, so in this, if you see the strategy, the for especially the type 1 strategy, which is for pasture. The strategy I was talking about, how it is done, the spacer is there, we have already shown the mechanism. So the interesting part about this is, this particular strain, that initial when was not named, now it is already named, so it is Thermoanaerobacterium virendrai. So the locus that we have available is we have in this type 1 and 3. So you can see in this particular native strain, missing. So whenever we are, is not giving me option. So I cannot think much about the Cas9. So we are not, as a strategy, first strategy, we are not thinking about heterologous expression, but we will be relying totally on our native CRISPR tools. So we will be first seeing how the native CRISPR tools are doing, and after that, we will take a decision whether we will be able to sorry. So if you see the availability, what is there in the genome itself? So type 1 is only having 1 copy. Type 3 is having 4 copies. So we will be, our strategy is, so right now we have started work on using type 1 as well as type 3. So there are 4 copies available with us. So we are starting with, we are giving like working simultaneously on both the types, and then we will decide later on, depending on the efficiency, that which will be a good toolbox. So since it is, the work is going on, so I'm not able to share much on what kind of things still be published. So type 1 and type 3, the PAM dependent cleavage of target is there. Okay, so in type 1, there is a PAM dependent, but in type 3, there is no PAM dependent. We don't need a PAM site. It is a PAM independent site. And it cleaves, so type 1 cleaves only DNA targets, 3 cleaves for DNA and RNA targets. And type 1 composition and organizing comparatively less diverse, and in type 3 composition and organizing comparatively more diverse due to gene duplication, deletion, domain insertion, and fusions. Type 1 is having 6 subtypes, 1A to 1F, and type 3 is having only 4 subtypes. And in type 1, DNA targeting can occur on both DNA strands, and in type 3, only template strand is targeted for function. So the advantages of type 1 and type 3. So with type 1, we are getting most frequent in CRISPR-Cas system, and PAM dependent, hence no problem of cleaving the self genome. So that is a good point with it, but we just have one to work with. And type 3, PAM independent, hence we can cleave targets lacking PAMs and can cleave both DNA and RNA targets. So this work is ongoing. So I hope when we meet sometime, we'll be able to share more on what is working for this system. So thanks a lot.
Yeah. Thank you. Am I audible?
Yes, ma'am.
Yeah.
Am I audible? Yeah. Okay, because I really have a very bad network. And thank you very much. Yeah. Thank you very much. Really, some information which was very important at this stage, because participants have been able to see actually a real-time work going on, and that yes, you will wait for maybe some more months and to see the outcome of the project, but definitely you had some important information about how you have started the work, how you have carried it over, and what are the conditions that had been problems, and so you had to change and go for a recombinating system. So this kind of shifting from the classical method to a different also is very important, and I think the audience have got some clear perception about how to address certain different strains, some different strategies, which we have been delving with for the last 5 days. So thank you very much, Dr. Bishers. Now, if there is any question from the audience, I will ask you to directly unmute, and you can raise your hand and just ask questions to Dr. Bishers. So I do not see at the moment, but I have some questions in between. If somebody is ready with some questions, please just you can type, or you can even unmute and ask. So Ranjita, while I got disconnected for my network issues, so I missed out certain points. So you mentioned some toxicity studies, means how are you managing? What I understand that your strategy will also have step where you need to check the toxicity parameters before you even accept. So what was that part? I did not get it because I missed that portion.
Actually, when this Clostridium thermosilum strain was worked upon, so toxicity assay, normally when we are doing the Cas9, so we know that when it has this inherent problem of not able to make the double cut in the DNA. So Cas9 normally introduces toxicity. So if we are using Cas9 that was used for this Clostridium, so that time the toxicity assay will be important. So in our case, where we are doing our in Thermoanaerobacterium, so in this particular strain, CRISPR-Cas toxicity assay, right now we have not listed it for our work because we are right now only focusing on type 1 and type 3. So there in type 3, like you know, there is no, in this, we don't have any Cas9 here. So we will be going ahead without the toxicity assay.
So what would have been your assay? Means how would you have tested it?
No, so in toxicity assay, we have this, so if you just actually, I will take you to that, just quickly share that strategy in the material method. So this is a published work. So in this, if you do this, so toxicity assay basically is used to demonstrate functional DNA cutting. Okay, so where the required, so the difference in the system requires, so we have two types of systems. So type 1B and your type 2. So in type 1B, you are going to put the spacer. So there is all transformations are done. And then depending on the number of cells killed, one equation is there. We put one equation. So in that equation, we will have the CFUs and no PAM divided by spacer, single guide RNA plus the CFUs of PAM divided by the spacer, single guide RNAs. And this whole system will be divided by CFUs in no PAM oblique spacer, single guide RNAs into 100. So when you put this formulae and whatever plate you have got on the plates, the transformed colonies, you are going to put all these values here. So that is going to tell us that what kind of toxicity level is there. So basically it is a kind of a calculation we are doing, trying to show that if you are involving this kind of your cell, see, when you have multiple options, like native options are many. So in our case, we have 1 and 3. How we are going to decide? Suppose in the Clostridium thermosilum, they have type 1 as 5 copies. So out of that, they had taken only 1, depending on all these essays and lot of background work. So this one published paper is covering of what they are doing. So depending on what transformant colonies you are going to get on the plate from various combinations, where PAM is there, where PAM is not there, a spacer is there, no spacer is there. So you are going to take it together and then see what is the toxicity of this system. So you want to take a system where your toxicity need not be very high, but you cannot avoid it. So suppose in this case, when the heterologous expression is taken from your this GeoS9, Cas9. So GeoCas9 is coming from a heterologous system. So definitely it will have
Hello?
And probably we are having a lost connection with ma'am.
Yeah. I think, yeah, she's joining.
Sorry.
No. We can understand.
Hello?
Can
you hear me now?
Yeah.
No, I cannot see myself.
Yes, ma'am.
Okay. So only so far my experience with CRISPR-Cas, I can say that since Cas9 is well defined and a lot of work is there. So people who are interested to work on it have a lot of literature and background to start their work. But when I'm working on a thermophilic system, so most of the groups in the world who are working in this, they are all just started and they have a lot of issues. So yeah, we are looking to design a system which is beyond Cas9. So Cas6, Cas3. So I have covered only a few things here. But there are other thermophiles also in which people are getting good result with Cas6. So yes, it's a very powerful toolbox. So like what you have conducted 5 days, we have seen that it is now applicable in, sky's the limit, it seems. It is applicable in all domain. So although in thermophiles we are struggling, but yeah, it's interesting to work on this.
Yeah. And Anuj raised Sahan. Can you ask your question, Anuj?
Am I audible, ma'am?
Yes, please.
Yeah, thank you, ma'am. And I have a question that because as we have studied in our classes that when there is a discovery about this or this invention of this genetically engineered organism like Pseudomonas for the oil spills, and there is a whole lot of debate whether it's okay to use that organisms in a natural ecosystems for the cleaning of that oil spills and all that. So I want to know that whenever you are working in these kind of systems, is it feasible to do this kind of work in the open ecosystems, or it is only for the lab-based experiments?
I think Jaya Madam will first answer on this.
See, actually, the both things are, you need to understand. If you have already heard from Dr. Kutubuddin earlier, like if you have to really go into the nature, then you would require a lot of regulatory bodies to pounce on you. Yes, it would be definitely encouraging to see all the new such strategies to be used, but I am really not the right person to answer, because the safety issues that are there, and whatever strategies that you apply, and you are going to apply it on nature, then you need to think about that, how safe and how much you are permitted. But yes, regarding microbes, maybe Dr. Bishash can highlight. But yes, you need to consider the surrounding, the safety, the spillage, whatever you apply new on in the nature, you need to think about how much you're permitted. So let in the lab scale, yes, you are always welcome, it's provided that you even you adopt all the safety issues. You cannot really be very safe with any organism. Always remember, you might think that this is a non-pathogenic strain or it is not going to harm, it is in our system. Still, working with any living organism needs a lot of conditions and rules to be abided by rules. But yes, Dr. Bishash will highlight, because she is working directly. She will have a better answer on this.
So, Anuj, this question, that whether or not. So if you see nature is constantly evolving. And it is evolving based on all kinds of signals, stimuli, pollutants, whatever is happening. So every living system on Earth is evolving. So that is a natural engineering. So, okay. So in natural engineering, nobody has a control. So we are not talking about regulations. But
yeah, natural engineering, definitely. Yeah.
Yes. So it is happening. We cannot stop. Nature is evolving. And now when we are doing at a human level, so whether we are talking about the GMOs or the SMOs, so there is always a concern, there is a lobby for and against, everything is going on. But then you cannot deny the impact of the genetic engineering, where we have such high-end therapeutics, and we could get out of COVID because of, the gift of genetic engineering and all the kinds of toolboxes that are available with us. See the vaccine, see everything, see the insulin, the life-saving drug. So it is directly engineered, and we are having a direct administration in the human body. So I'm not denying that we need regulation. Regulations are very much in place, but we have to work. We cannot stop. We have to design things, and we have to see that the microbes, and since the tools that are available now is very advanced, whether you're talking about all the omic toolboxes, you are talking about all the various interdisciplinary information we have available with us. So it is not that difficult now to know what is happening and whether it is having a contact or it is harmful for some kind of overflow into the environment. We can track that very easily. So even the industries who work on recombinant microorganisms, they have stringent quality control, stringent strategy to see that containment is very good. So we cannot stop work. There are a lot of work remaining. So how this COVID vaccine was possible in such a short time? See the dramatic effect of that. So everybody was so scared, that this third peak will be disaster for India. But see, so many countries are still suffering. But then in India, okay, we have a good immune system, I guess. But the gift is from the vaccine, and it's all because of this recombinant engineering technology that we have. So Anuj, since you are a student, you will be working in this field. So just keep this in mind. We have to have a balance. So we cannot sit, blindfolded and fold our hands and say we cannot do genetic engineering. Nature is doing that, and we are also a part of it. So we are also doing it. So let's be more conscious, and let's do it with a very good scientific conscience. That's all I will say.
Yes, absolutely. Absolutely.
Thanks for your advice, and thank you so much, Jai ma'am, for giving an opportunity to ask the questions in the whole FDP program and all that. Thank you.
Oh, yeah. My pleasure as well. Okay, and since we just have a few more, just one last question. This is a technical question, Ranjita. I do not know whether others would have the same. Because since the network, I had network issues, you had been selecting colonies based on wherever, means whether you have done it or not, finally, I don't know, but when you are selecting colonies based on which has got the editing and those which are non-targets, how are you selecting between these two? Just on plate. I just need to know the technique.
See, for this particular case that we are discussing here, so there is one mutation that is being introduced in the PYRF gene. Okay, so this PYRF gene is, if it is mutated, then you can easily screen in a plate which is having 5-FOA, that is an analog of uracil. So we add this chemical on the plate, and if it is successful, you will get to see. If it is not successful,
okay.
I mean,
so that means the way we do it.
We have yes. So for all the system, you have to find something that you can, and yes, that is not going to interfere with the metabolic cycle of the system, as well as it is able to give you some selection marker for the plate.
So that means the taking this idea, means this principle, I think your the two plasmid strategy follows the same principle, right? Because the first one has to be taken up, and then only the second one will be able to incorporate, right? So I was just not trying, I was getting confused because I could not hear the complete thing. So we are almost towards, we are in fact at the end of this last session. And I think all the participants have attended all the 14 sessions. It brings really great pleasure for me to have arranged this FDP with all the support of our distinguished speakers, and also Ranjita ma'am is there. Thank you very much for your precious time. And it was actually, I want to share with all that when this proposal had been was brought forward to me from our Honorable Vice Chancellor, the first person at that moment, actually, I just called Dr. Bishash, our Ranjita ma'am, and I just said that we have to hold one FDP, and I have to organize this. So what is your say on it? She said, yes, ma'am, go ahead, and I will be happy to take care of one session. So that was how I started off, with the support from my colleague, Dr. Ranjita Bishash. And then I took it over to the rest of the speakers. And of course, Debojyoti was one who really helped me, and the rest is all history. So with this, I thank all the participants to attend all these sessions. And also I thank, and on behalf of our department and the university, I thank Ranjita once more for her precious time. She nicely explained the project that she is taking care of. She has immense funds on this project, DBT project. She has crores of rupees on this project. So we are very proud that, and I wish her all the very best with her students and her team, that she gets very good results, very promising outcomes, so that we can be proud to share in some way or the other to the country and to the world. So with this few words, I take this opportunity to end this session. With your permission, Ranjita, we will now stop. We will bring an end to this particular pre-lunch session. And before I have some announcement to make to the